just want to start off by, as representing the PheoPara Alliance, and in cooperation with the NETRF, we're providing travel awards for those who are interested in promoting PheoPara research. The PheoPara Alliance, just a quick introduction, is a patient-directed advocacy group with a small research promoting arm. I'm representing them as part of the medical advisory and research advisory boards. And there's the website. And then specifically, we wanted to recognize 2 investigators who are actively pursuing research in this area. Later, you'll hear from Hector Gonzalez-Cantu about his work in TMEM127-driven pheochromocytomas. And I'd also like to recognize Sarah Galak, who is doing modeling of pheochromocytomas from dogs and has developed an organoid system. So their work has been recognized with a $1,000 travel grant, which we're very happy to provide, and congratulate them on their work. So thank you. So with that, we'll get started with the next session, and I'll be calling up Dan Merrick. Is he? Oh, come on up. Who's going to solve all our questions about DIPNEC in about 10 minutes.
I get to go after lunch, after the pictures, and I got to solve all this. This is tough. All right, well, I'm grateful for the opportunity to present our work today on whole exome sequencing of premalignant invasive neuroendocrine lesions and diffuse idiopathic neuroendocrine cell hyperplasia, otherwise known as DIPNEC. Just a small disclosure, no relation to the data I'll show. And then this has already been summarized pretty well today. So let's move on to the second slide, which really focuses on the area that we're particularly interested in, and that's the premalignant lesions. For the more aggressive neuroendocrine type tumors, they're really, for the most part, are not well known or no known existing premalignant lesions. But neuroendocrine cell hyperplasia, or NEC lesions, are well-recognized unique lesions that are associated with development of carcinoid tumors in the lung. These premalignancies are often seen near carcinoid tumors, but then specifically in patients with DIPNEC, they're seen throughout the lungs, and numerous lesions are seen. However, there are other disease processes in which they can be found that are not malignant, and so it's thought that malignant potential is not always associated with these lesions. So DIPNEC was first described just over 30 years ago, actually in an article by my longtime colleague, York Miller at the University of Colorado, who's also a co-investigator on this particular project. It's characterized by multiple bilateral foci of neuroendocrine cell hyperplasia. And specifically, these really are focused on the medium and small airways of the lungs. And ever since that description, we've actually kept pretty close track of any sort of pathologic features of DIPNEC in resected specimens at the University of Colorado. So York has gone through and characterized for our 54 patient cohort the clinical signatures that we see in patients with DIPNEC, and that includes a persistent cough, a progressive dyspnea, and respiratory compromise that can actually lead to respiratory failure, sometimes necessitating lung transplant. There's also characteristic radiographic changes, multiple nodules, and air trapping. And so we've used all of these different characteristics here to classify each of our cases as to whether there's strong evidence of DIPNEC, intermediate or weak evidence of DIPNEC. And so that's shown in this table over here. These are the 54 patients that we have in the cohort. We used a 2-point scoring system for pathologic and 2 points, up to 2 points for clinical radiographic changes. And you can see that a little over half of our patients have characteristics that fit into the strong DIPNEC category. And when we look at some of the associated changes, some of these really fit well. It's known that Dipnic develops at a younger age than sporadic carcinoids, and we can see the difference there. There are multiple nodules are more common and bilateral in this group, although there are some inconsistencies. For one, Dipnic is known to be very strongly associated with female sex. There isn't any difference here. So I actually will, as I go through the rest of the slides here, talk about, for this category here, moderate and weak evidence Dipnic cases. So our project is exploring the hypothesis that Dipnic results from a somatic mutation affecting the pulmonary neuroendocrine cells of the airways, leading to their proliferation and potential clonal wide dispersal throughout the bronchial epithelium. And so Hoy Yu, an instructor in our department and an important person in our laboratory, has really been leading this project. What we are doing is specifically looking for multiple lesions that we can sample and do whole exome sequencing within the airways of each patient. With those, we microdissect those, extract DNA, and then we also microdissect either lymph node or normal lung tissue to use as reference. What I'll talk about today is mainly specific aim 1, although we will have some data on some of the carcinoids associated with this. Our specific aim 2 will ultimately compare the carcinoid tumors from Dipnic patients to those with a sporadic carcinoid tumor. And ultimately, we want to identify whether there are some somatic mutations that lead to Dipnic and whether there's a clonal distribution of these premalignant lesions, ultimately, hopefully leading to potential therapeutic approaches or prognostic tests. So for the Dipnic lesions, for the neuroendocrine cell hyperplasias, we've been collecting those from locations that are remote from the actual invasive tumor when that's present in these patients. In general, we're trying to use just a single neck lesion for each of our DNA samples, but occasionally, if they're very small, we combine them. And these are the histologic characteristics here. You can see it is kind of subtle, but there is this expansion of the basal layer by neuroendocrine cells, which you can recognize on H&E. They do have some of the characteristic changes, the salt and pepper chromatin, et cetera. But then you can see on a synaptophysin stain, there's a significant, in the normal airway, there'd be 1 or 2 individual synaptophysin positive cells. And then in the small airways, you can see this is another characteristic. This is almost completely occluded. The asterisk there is just the one spot where there's still some luminal area left. Eventually, then these can progress so that they invade. And so this would be a tumorlet. Once it gets bigger than 0.5 centimeters, then we call it a carcinoid tumor. So in this preliminary analysis, we've done whole exome sequence on 19 of our 54 patients. You can see the breakdown of whether there's strong, intermediate, or weak evidence for actual Dipnic. And you can see over here, we are able to get multiple neck lesions from almost all of these. For some of them, we only have one. We generally have a tumorlet that's associated with it also. So in this ongoing analysis, what I'm really going to talk about today are just some analyses looking at for single nucleotide variants and small insertions and deletions. So this has been a relatively challenging area in terms of getting sequencing information, but fortunately we've had a lot of experience with this, with bronchial dysplasias, the precursors to squamous cell carcinoma. You can see they're about the same size and about the same cellularity. And so in the lab also, in addition to Hoy actually going through and finding all of these lesions and microdissecting them, Nate Pecos has been in charge of our DNA extractions. We've used a technique where we employ carrier RNA. And when we initially did this with bronchial dysplasia, we were able to show, I don't want to go too far into this, but for very small lesions, the carrier RNA does at least double our yield here. And when we look at whole exome sequencing with and without carrier RNA, the metrics are at least as good with the carrier RNA. So these are the sequencing metrics from the 19 cases that we have looked at, and that includes 51 actual lesions and then the 19 reference specimens. So you can see that we actually have pretty strong mean coverage in all of these lesions. The percent duplicates, the blue bar is actually the ones with the low percent duplicates, so pretty good in that respect, and not too bad in terms of off target, which is generally 25% or less. So we've approached this with two sort of approaches in terms of trying to determine whether or not we have a significant somatic variant that's associated with the development of these lesions. First thing we did was to try and look for a specific variant that was present in more than one of our patients. When we did this, we used some relatively lenient filtering where we just wanted the variant to be less than 20% in our reference specimen, and then at least twofold higher in the lesional versus the reference specimen. When we did that, we found 113 specific variants that were in 2 or more cases. For the purposes of today, we just looked into the 18 that were in more than 3 cases. Unfortunately for these, a significant number of those were actually artifactual variants, predominantly identified because of a number of duplicate reads. But there were two variants that have good-looking sequencing metrics. One is in CMPY in the promoter region, and one is in TALEN2 in the intronic region. And when we look at those, we can see over on the left-hand graph here that the majority of those are in cases where they either had strong or intermediate evidence of being DIPNIC cases. For the CMPY3 variant, all of the lesions in all of the premalignant lesions in those 3 cases showed that particular variant, whereas only one of the 2 did in 2 of the 3 patients that had the TLN2. And then finally, when 5 of these 6 cases had carcinoid tumors, and you can see here that 3 out of the 5 had the same, of the invasive tumors had the same variant as we found in the neuroendocrine cell hyperplasias. So we took another approach then following that by actually looking within patients to see if we could find by genes whether there were particular genes where we found multiple variants. And so again, we applied the same filters basically and ended up with a list of, I believe it was around 6,000 variants. To narrow that down for today's purposes, I actually used some previously published from targeted assays, some previously published genes that have been associated with alterations in lung neuroendocrine tumors, and then went through, and if we found a particular gene, then we'd see if there were other patients that had variants in that gene also. And so using that technique, we have about 122 variants, and we thought about 50 of them were strong candidates. We're maybe about halfway through, or a little more than that, evaluating these lesions, these particular variants. And those are summarized in this table here. So you can see there are a number of different genes that were involved here. Here we really just restricted this to looking at alterations that were in the coding region or in the splice site regions. You can see over in the columns on the right here that all of these were present in at least 2 lesions and up to 4 lesions, and a couple of them were also present in multiple patients. And so of those, the CMSD3, well, I think BRCA and MSH6 are genes we're all familiar with as being involved in DNA repair. The CMSD3, this is one that was previously published as being mutated in lung neuroendocrine carcinomas, and does show some role in dendrite development. For the PRAMEs and the MBF factors, there's some variety of different alterations that we see here. You can see that the PRAMEs are associated also with neural processes and associated with alterations in a number of different tumor types, and has sort of some classic oncogenic sort of properties, a negative regulator of apoptosis, positive regulator of proliferation, commonly overexpressed in tumors. And then MBPF genes that showed some variants are from what is known as neuroblastoma breakpoint family. It's been associated with fusions in neuroblastoma, and then also appears to have some tumor suppressor activity in other tumor types. So just in conclusion then, in this interim analysis, I think we've shown that microdissection-based analysis of these neck lesions can provide adequate DNA for whole exome sequencing in most cases. We may see some early evidence of discrete multifocal clonal neck lesion hyperplasias with possible candidate somatic variants within and between patients. And so I think then the additional analyses are going to be very important for this particular analysis. I think applying the appropriate filtering algorithms is going to be important for us to be able to find the potential driver mutations in these lesions. And of course, this is only about a third of our cohort, and so we hope to see more data in the future analyses. And so with that, I'll just close. I think most of the people, should mention Thomas Danhorn as our bioinformatics person. We've had a lot of support that's helped us learn how to work with these limited premalignant lesions in the lung. This particular work that we just discussed here has been completely supported by the Neuroendocrine Tumor Research Foundation, and we're appreciative of that. Thanks.
Okay, and so we'll open up for some questions.
Multifocal small intestinal neuroendocrine tumors linked to zones of oscillating IGF-2. Can you look for, which I know it also occurs in lung, can you look for oscillating IGF-2?
Thanks for the suggestion. We'll definitely keep IGF-2 in mind.
Jules Derks from Maastricht, thank you. Very interesting study, I think, and very relevant to evaluate. What I was wondering, because we had thoughts of this as well over the past few years to do such research, how sure are you that you're evaluating neuroendocrine cell hyperplasia lesions, and how did you make sure you're only measuring this lesion? Because it's quite difficult when you cut through these specimens, actually 4 slides further, 4 microns, it's already out.
Yeah, so we did do microdissection, and we do interim H&Es to follow those. And so it's an excellent point. We did end up combining some lesions because we frequently are trying to get a dozen or 15 levels for microdissection. And there definitely are lesions where it's gone by the time we get to the second H&E, on level 5 or whatever it might be. So that's, what we're trying to do anyway to enrich for the actual hyperplastic.
But it wasn't laser dissected. It was really just pipetting or just cutting it out from the FFPE tissue on the original slides, I think.
Yeah, exactly.
Okay. And then the second question, you evaluated germline mutations, I think, using lymph nodes and normal lung tissue.
Are you getting at, could there be a polymorphism or something like that that's associated with DIPNIC?
Yeah, well, what I personally am wondering is, because it's so spread throughout the lungs, why is it a somatic mutation that we're targeting? So why is it somatic? What is the mechanism behind DIPNIC? So is it a germline variant, which causes these patients to have all these multiple lesions? So maybe the BRCA1. But then the question is, how did you make sure you're only looking at somatic, and didn't you find some germlines maybe in your data?
So we used the lymph node sequence as our reference sequence. So anything that was present in both, then we actually eliminated. I agree with you, and I think that the data from the lymph nodes and from the normal tissue actually could be valuable in looking for a polymorphism. The reason we actually hypothesize this is we have seen, it might sound a little strange to think at multiple sites around the lung, you've got the same driver mutation, but we've actually seen that for the precursors for squamous cell carcinoma and bronchial dysplasias with the P53 mutation that's present in all of the different sites. We've seen that in work we've done, but that's also been done by the Janes Laboratory in London, where they had this great study where they showed the same clone actually moving to the other lung over time. And so that's kind of where we started. That's the rationale we used anyway.
Okay, thank you.
One more question.
And why did it have to be genomic and not epigenomic?
Good point, and that's been something that's been brought up. And I think there's a number of different possibilities for what might be driving this. And so, yeah, we just started looking for actual somatic genomic mutations.
I think we'll transition to the next section, but since this is the PPGL is mentioned here, I think it's interesting parallel to Carney triad, which is predominantly female and is an epigenetic and has multiple locations, so an early embryologic epigenetic event, and whether or not there's parallels here. Okay.
All right, so the next speaker is Hector Gonzalez-Cantu, and will be telling us about TMEM 127, exerting a tumor suppressive role in pheochromocytoma by mediating RET ubiquitin-dependent degradation.
Right, so I'd like to start by thanking PheoPara Alliance for the Travel Award and NETRF for the space to talk about my work. And sorry, so some disclosures. And as Justin mentioned earlier, my work deals with pheochromocytomas and paragangliomas, and these are catecholamine secreting tumors. They are mostly benign, although a percentage of them can metastasize, and these are driven by mutually exclusive driver events, and approximately 40% of them can be germline. So the function of these driver genes is only partially known, and this is currently a gap for proper diagnosis, surveillance, and treatment of patients having these tumors. And the focus of my research is on the poorly known tumor suppressor gene, TMEM127. And TMEM127 codes for a transmembrane gene. This is ubiquitously expressed in vertebrates, and it confers susceptibility to PPGLs due to pathogenic germline loss of function mutations. But its function is relatively unknown. What we have shown is that compared to wild-type TMEM, mutant TMEM seem to lose its wild-type distribution, which suggests to us that part of the tumor protective role of TMEM lies in its endomembrane trafficking properties. But again, this is just an observation based on what we see in its distribution, which really motivated us to ask, how does TMEM protect against pheochromocytoma? And PPGLs, they associate in 3 molecular clusters. TMEM, they cluster in the cluster 2 or the kinase cluster, which is characterized by a kinase signaling signature. So is RET, which is a receptor tyrosine kinase that is also a driver of PPGLs. So this molecular association led us to ask if TMEM and RET were perhaps mechanistically related. And to address that question, we looked transcriptionally, and we saw that TMEM and RET mutant PPGLs had no differences at the transcriptional level, which led us to proceed into the post-transcriptional differences. And we saw that PPGLs harboring TMEM mutations displayed high levels of RET protein expression and high levels of RET phosphorylation, suggesting that there was RET accumulation and RET hyperactivity in these tumors. And this was not shared with PPGLs harboring other driver events. And this was also shown by staining in IHC, where TMEM mutant PPGLs had high levels of RET. We expanded these findings into a cell line where we showed that TMEM loss was reproduced, the RET accumulation in cell lines, and this was conserved in mice, where TMEM loss also resulted in high RET levels. We then looked at what was the role of the functionality of TMEM in the RET accumulation, and we showed that a pathogenic TMEM mutant failed to suppress RET to the levels of wild-type RET, suggesting that the RET levels were dependent on TMEM functionality. And we also looked at RET clearance with a cyclohexamide experiment, and we showed that TMEM depleted cells had a decreasing RET clearance, suggesting that TMEM had a role in RET stability at the protein level. Right. So in an earlier talk, they discussed endocytosis, and this is very relevant when talking about cell surface receptors. So we were also interested in looking at what role TMEM could have in regulating RET's internalization or endocytosis. And we showed that in TMEM depleted cells, RET was translocated to the cell surface, and this was not seen in a TMEM wild-type cell. And we showed that this was reproducible in a different cell model, so these are HeLa cells. And further, we showed that reconstituting these cells with a wild-type TMEM reversed, or I guess recovered RET's wild-type distribution, but another mutant TMEM failed to, I guess, rescue the effect. Again, supporting the fact that functional TMEM is relevant for not only RET stability at the protein level, but also RET distribution. And we also showed that in TMEM depleted cells, RET colocalized less with an early endosome marker and with a lysosome marker, suggesting that TMEM not only regulated RET's distribution, but also its association with the endosome system and the lysosomes. So we were then interested in looking at which motifs in TMEM could perhaps be governing this regulation. And TMEM has two C-terminal PY motifs, which canonically interact with WW domains. NETRF, which is an E3 ligase of the ubiquitin system, was previously reported to target RET for ubiquitination by Lois Mulligan's group. And we were interested in exploring whether if TMEM and NETRF had any relationship in this process. And indeed, we showed that a PY mutant TMEM, where we mutate these motifs, failed to suppress RET to the levels of the wild type, suggesting that these motifs were relevant for this regulation. And we also showed that the PY mutant TMEM also resulted in less RET colocalization with LAMP1, suggesting that these motifs had some relevance for this regulatory mechanism. Furthermore, we showed that TMEM interacted with NETRF in a PY-dependent manner, so the PY mutants failed to interact with NETRF, and the TMEM was needed for the RET-NETRF interaction, which built our model so far that TMEM and NETRF interacted in cells to target RET for downstream events that were TMEM dependent. And intriguingly, a separate model, in a Salmonella infectivity model, they had also seen that TMEM played a role in regulating the major histocompatibility class 2, which is also a cell surface receptor, by associating with WWP2, which is in the same family of ligases as NETRF. So this really motivated us to look at some potential parallels between their model and our model. And indeed, we showed that RET ubiquitination was influenced by TMEM in a PY-dependent manner, and that this was NETRF dependent. So at this point, our model led us to further, our model here showing that TMEM recruited NETRF to target RET for ubiquitination. And as a tumor suppressor, we were also interested in looking at more direct oncogenic features of our model. And we saw that our TMEM depleted cells had a higher proliferative rate, and that these cells were sensitive to sulpercatinib, which is a RET kinase inhibitor. We generated double knockout RET and TMEM, and we showed that these cells were not responsive to sulpercatinib, suggesting that this effect was RET dependent. And transitioning to an in vivo model, we xenografted these cells in mice, and we saw that the proliferation recapitulated the in vitro observations, and that these cells were also responsive to sulpercatinib, as you can see here by the tumor burden. So this data suggested to us that TMEM loss in these cells accumulates RET, and this is a factor that drives oncogenicity in this model. And overall, our data shows a mechanism of suppression whereby TMEM antagonizes RET, and this is due to TMEM's ability to recruit NETRF to the cell surface to target RET for ubiquitination, leading to its endocytosis and lysosome-mediated degradation. In a TMEM loss-of-function state, or where TMEM is depleted genetically, RET accumulates in the surface as it is not targeted by NETRF, and RET signaling drives proliferation in these cells which is therapeutically sensitive to inhibition. And just like to thank the people in our lab. So this is a big group that we're very proud of our work. And I'd also like to thank the funding entities that make our work possible and the patients who generously donate samples for us to study. Thank you very much.
Great talk. We have time for a few questions. We'll go there first and then.
Great talk. Good thing you sponsored. Great investment. So do you think this applies to all TMEM? To all the tumors?
All TMEM tumors?
Yeah.
So there are different mutations in TMEM.
Right.
The one factor is that they all lead to PPGLs. We haven't explored if all TMEM mutants are applicable in this model. It's something we're considering. So what we do know is that different TMEM variants seem to impact its distribution differently. So that may be a factor that differentially regulates RET, and it would have implications for the RET dependency of those cells. So we're still trying to address that question. It's possible that only some TMEM variants influence RET in this model.
I mean, because if it were, and it might even apply across tumors other than pheoparis, any tumor that would have a TMEM mutation could be selpercatinib vulnerable, for example, right?
Right. So really, I think the literature supports TMEM as a tumor suppressor for pheos only, but definitely if a cell expresses RET and TMEM is lost in those cells, then it's possible that RET could also be disturbed in those cells.
Mm-hmm. Okay.
Hi, Hector. Great work.
Thank you.
I was wondering whether this is specific for RET, and whether you're planning to do cell membrane proteomics to see maybe you have other receptors that are sitting in the membrane and are affected by TMEM mutation.
Yeah, so that's a great question. We focused on RET because our primary samples, it was something that we saw, and RET and TMEM share some features clinically as well. But Lois Mulligan published a beautiful paper recently where they showed that TMEM loss altered membrane dynamics. So it is possible that TMEM regulates other cell surface receptors by similar mechanisms. It's something that we're interested, but we haven't explored fully. So there might be other targets that are also regulated by TMEM.
Thank you.
Thank you.
Okay, great. I think we'll continue the discussion during the discussion panel part. Thank you, Hector. So up next is Minah Kim from Columbia University, and she'll be presenting on vascular regulation of liver metastases in PanNET.
Okay. Thank you, Justin and Don. This is my fourth time attending this symposium, and it's always a great honor to be here. And it's been, 4.5 years since I started my lab at Columbia, so I honestly feel that I'm growing in support from NETRF, which is great. And I'm very happy to share our recent progress and ongoing work here today about vascular contribution to liver metastasis in pancreatic neuroendocrine tumors. Okay. So previous studies have shown that anti-angiogenic treatment not only causes selective regression of immature blood vessels, but also improves vascular integrity and function, which increase vascular perfusion and consequent drug delivery to the tumors. But not only this, that anti-angiogenic treatment can reprogram immunosuppressive tumor microenvironment. So recent clinical trials consistently for some types of tumor like hepatocellular carcinoma and RCC have demonstrated a beneficial efficacy when checkpoint inhibitor was combined with anti-angiogenics, especially bevacizumab, anti-VEGF. So all these recent clinical and preclinical studies have emphasized the importance of understanding the immune and vascular crosstalk, and also the targeting tumor vasculature to improve immune suppression in the tumor microenvironment. And here, the unmet need is to better understand the mechanistic contribution of anti-angiogenics to immune activation and also clinical response, which is the main question that my lab has. So as you know, the pancreatic neuroendocrine tumor patient, they frequently develop liver metastasis, and the targeted therapies are limited to control this metastatic disease. So the main question that we have here is how vascular destabilization contribute to immune microenvironment in PanNET liver metastasis. So So previously, I could share this data where we profiled anti-angiogenic genes in the human PanNET liver metastasis comparing normal livers, where we found multiple angiogenic genes, upregulated, downregulated, and recently we could compare these angiogenic genes in pancreatic tumor, in the primary tumors as well. And interestingly, we found for certain genes showed similar patterns of upregulation and downregulation, like angiopoietin 2. However, we also found the the pattern of expression, for example, the PDGF and the VEGF-C, suggesting differential vascular regulation between metastases and primary tumors requiring further investigation. So anyway, the angiopoietin 2, the molecule that we focused on, showed one of the most unregulated genes in both in liver metastasis and then primary tumors. So briefly, I wanted to introduce about angiopoietin 2. So the angiopoietin 2, together with the VEGF, stimulate angiogenesis. But more importantly, this angiopoietin 2 is a potent vascular destabilizing factor as a type 2 antagonist. And angiopoietin 2 usually stored in endothelial cells, unlike VEGF. The VEGF is expressed by tumor cells or other types of immune cells, but angiopoietin 2 is stored in endothelial cells and can be released in many pathologic conditions upon inflammatory stimuli. And we often see this angiopoietin 2 upregulation in many types of tumors. And here, the angiopoietin 2 can suppress type 2 receptor and consequently induces vascular leakage by impairing pericyte coverage and also the endothelial junctions. Although the angiopoietin 2 is often unregulated in tumor, but the role of angiopoietin 2 in immune evasion of tumor microenvironment is largely unknown. So this is the question that we had for the PanNET liver metastasis. So to understand the role of angiopoietin 2 on liver metastasis, so we mainly used RT2AJ mice models. So we really like this model because we can really study the late stage of liver metastasis. So, and also to block angiopoietin 2, we have been using very selective anti-angiopoietin 2 antibody by collaborating with Regeneron. So we block angiopoietin 2 in two different time points. One is more like early metastasis. The second is the late stage of metastasis. And interestingly, in both time point, in early and late stage, we could find that angiopoietin 2 inhibition significantly reduced metastatic burden in the liver consistently. And if you see this graph, the black is control and red is anti-angiopoietin 2, and then green is onset of treatment. So we can see this angiopoietin 2 inhibition induce a disease stasis in the liver metastasis. So to understand mechanism, we first check vascular stability. So because we know the angiopoietin 2 induces vascular destabilization. So we wonder this angiopoietin 2 blockade can normalize and stabilize blood vessels of liver metastasis. So we look at the vascular leakage by staining extravasated fibrin, and we found angiopoietin 2 significantly reduced vascular leakage of liver metastasis. And additionally, we found this angiopoietin 2 inhibition can increase pericyte coverage on endothelial cells, but also improve the endothelial junctions in the liver. And the recent studies have shown that the improved vascular function is important for immune cell infiltration to the tumors and improve immune suppression. So we are wondering whether this angiopoietin 2 inhibition and improved vascular function can affect T cell density and function as well. So we look at first the CD8 T cell density in the liver metastasis after blocking angiopoietin 2. And we found the angiopoietin 2 inhibition significantly increased the number of T cells in the liver for both CD8 and CD4 T cells. And interestingly, not only the density of T cells, but also we found angiopoietin 2 inhibition increased the frequency of activated CD69 positive and granzyme B positive T cells. And not only the liver met, we recently checked the primary pancreatic neuroendocrine tumors, and we found first, if you can see the black dots, which is control livers, and where we can see much higher number of CD8 T cells at the tumor periphery compared to the tumor core, and angiopoietin 2 inhibition significantly increased the T cell numbers in both both tumor periphery and core. So to understand the mechanism by which angiopoietin inhibition improves T cell infiltration, we look at adhesion molecules and chemokines responsible for T cell infiltration, and we found specifically the CXCL9 was unregulated after angiopoietin 2 inhibition in the liver metastatic tissues. And now, as a current work, we are further trying to understand the mechanism differ using the multi-omic approach, which is current work. The other thing is that we also look at the myeloid cells beyond T cells after blocking angiopoietin 2, especially the TEM is tied to expressing monocyte, which can be directly regulated by angiopoietin 2. But we found that the TEM proportion in the immune cells in the liver metastasis of pancreatic neuroendocrine tumors was very low, so it's kind of hard to compare. For the other cells, we found no big difference between control and anti-angiopoietin 2 for M1 macrophage or dendritic cells or monocytic MDSCs. However, we found significant reduction of M2 macrophages after blocking angiopoietin 2, which was quite interesting as well. And also, we tried to demonstrate the load of T cells for in the anti-metastatic action of angiopoietin 2 inhibition. So we did two separate studies. So one is angiopoietin 2 inhibition in immunodeficient mice, and second is this experiment is CD8 depletion study along with the CD4 T cell depletion. So here, both studies support the importance of CD8 T cells. And here, if you look at this CD8 depletion group with angiopoietin 2 inhibition, we can see quite comparable high metastatic burden with similar to control livers. So it really said the load of CD8 T cells in the anti-metastatic action of angiopoietin 2 inhibition. And we can kind of see the partial effect after CD4 depletion, suggesting the complementary role of CD4 T cells. So I hope we convinced that this angiopoietin 2 blockade suppresses the growth of liver metastasis from pancreatic neuroendocrine tumors by promoting T cell recruitment, which was accompanied with improved vascular integrity. And this recent work was just published last month. And as ongoing work, as I said, we are currently working on investigating the mechanism deeper by which angiopoietin 2 inhibition promotes T cell infiltration, but also function in PanNET liver metastasis using multi-omic approaches like single-cell RNA-seq and also the spatial transcriptomics. And the other interesting project that we are working on is to assess the potential of vascular leakage of primary tumors in predicting immune status, metastasis, and prognosis among PanNET patients. So we acquired multiple primary tumors now, so we are about to look at this phenotype of vascular leakage. But for the preliminary data, when we look at the melanoma primary tumors, we found very interesting heterogeneity for the vascular leakage. So we are wondering about how the potential of this vascular leakage as a prognostic biomarker in PanNET patient. The last thing is that the SOPI presented yesterday, so we are working hard for identifying the load of VEGF and also the impact of VEGF inhibition for long term, when we treat for long term. So here, So here we found that in the liver, when we did VEGF inhibition for 1 week, we found dramatic the effect in terms of the liver met burden, but also vascular density. However, when we extended our treatment over 5 weeks, we kind of see the rebound angiogenesis and the liver metastatic burden, and we try to understand what's going on there regarding also the immune aspect. So the SOPI is characterizing the endothelial and immune phenotypes for this project. Okay, so thank you so much for my lab members and very supportive collaborators, and also the support, especially for the NETRF funding. Thank you.
So while we're waiting to take questions, maybe I can just get a quick one in. You said you saw increased T cell infiltration. You're just kind of doing numbers. Have you done anything to characterize proliferation, T cell receptor, evolution, things that show reactivity?
No, that's our future experiment. We didn't do that yet, but we wanted to see, the more like T cell phenotype, the function, and also exhaustion, and further characterization of T cells, but we didn't do that yet. Thank you.
Hi, lovely talk. It's interesting because back in 2009, we showed that you could measure angiopoietin-2 in the blood, and that in pancreatic neuroendocrine tumor patients, and that correlated with liver metastasis. It also correlated with burden of disease as well. So it's actually quite a good biomarker in its own right, and it might be interesting then to tie that in as well with your model.
Thank you. Thank you for the comment.
Hello, great talk, congrats, and very intriguing concept.
Thank you.
A couple of questions. First, which kind of antibody are you using against angiopoietin? I mean, which class, IgG1, IgG2, etc.? And have you considered that there is a chance that when using immunoglobulins, you may activate the immune system if they are recognized by immune cells, and that, like for example, might explain some. I'm not saying that is the what's going on, but I mean that might contribute to explaining some data. The other comment is, have you explored the spatial distribution of T cells? Are they close to the vessels? Are they far away from the vessels? I think that might be really intriguing in order to understand the effects of angiopoietin inhibition.
That's a very good comment. In terms of the T cell distribution in the tumor, we usually see the higher density of T cells at the tumor periphery, where vascular density is also higher. So, but that phenotype is more clear in primary tumors. For the liver metastasis, we didn't really specify the T cell numbers at the tumor periphery and the core. And also, we didn't really correlate with the vascular density, but that's a very good comment. Thank you.
And what about the antibody? Do you know the class of the antibody?
No. The antibody that we use is anti-angiopoietin to the Regeneron 910. So we are collaborating with Regeneron. So it's a selective antibody, because some antibody target both angiopoietin-1 and angiopoietin-2, but this is a selective antibody for angiopoietin-2.
Amina, great work. Congratulations. I think the group of Gabriel Berker published a paper in 2019, I think, in which they used the RIPTAC model, but it was the Black 6, which is the insulinoma, and they show when you do anti-VEGF and with anti-PD-L1, you get these high endothelial venules or tertiary lymph nodes. So I wonder whether you've seen any of these tertiary lymph nodes in your model, if you're considering to do combination with PD-L1, and what are your thoughts if you had to choose between VEGF and angiopoietin-2, which one would you choose?
Okay, so your question is first look at the lymph node.
Whether you have tertiary lymph node formation, either in the primary tumor or in the liver.
Yeah, that's very good comment. We didn't. Yeah, we focused on liver mets so far, but I think it's very good idea to compare the lymph node metastasis as well as a readout. The second question is about, can you repeat it?
If you combine the anti-angiopoietin-2 with anti-PD-L1, would you see even a better response? Yeah.
You mean the checkpoint inhibitor?
Yes.
Oh, okay. So in our model, when we tested, not PD-L1, but when we tested anti-PD-1, there was a resistant. So when we block angiopoietin-2, I saw the dramatic anti-metastatic effect, and anti-PD-1 alone treatment was resistant. And when we combine this anti-PD-1 and anti-angiopoietin-2, we saw the very similar the burden to anti-angiopoietin-2 monotherapy. So we didn't really see additive or synergistic effect at all.
Okay. And do you have a sense about inhibiting angiopoietin-2 versus VEGF, which one will be better?
Well, that's another question, but I think the other groups, they didn't really focus on liver metastasis, but the primary tumors, I think you know better than me, but they show them blocking angiopoietin-2 and VEGF together, they have a synergistic or better effect, at least in the primary tumors.
Yeah, great. Thank you.
Thank you very much, Minah, and for the great questions. We're going to move on to the next speaker, Scott Oakes, who's going to tell us about mTORC1 ATF4 signaling driving amino acid biosynthesis in pancreatic nets.
Great. Thank you very much. It's great to be here again. This is always one of my favorite meetings each year, and it's good to see so many old friends. Figure out how to use this thing here. Let's see. Am I doing? Oh, the big button in the middle. Okay, so we're revisiting mTOR signaling a bit, and as a reminder, we know that TORC1 is hyperactivated in most PanNETs, and that the TORC1 inhibitor everolimus was FDA approved over 10 years ago, but it's for PanNETs, but its benefits are pretty modest, and patients quickly develop resistance. And so some of the big questions we're interested in is really understanding how does mTORC signaling promote PanNET growth, and then how do PanNETs resist mTORC inhibition? So first I want to talk a little bit about the integrated stress response, because this is going to be relevant here in a minute. This is an ancient defense mechanism that cells use to adapt to a wide range of different stresses. It is triggered when one or more stress-sensing kinases like PERK, GCN2 detect damage, and then they phosphorylate EIF2 alpha. When EIF2 alpha is phosphorylated, it suppresses cap-dependent protein translation globally, but then ATF4, because it has internal ribosomal entry site, becomes a privileged transcript and is highly translated in this scenario. And ATF4 then upregulates a whole bunch of amino acid uptake mechanisms, synthesis mechanisms, rewires metabolism, promotes homeostasis. And together, these outputs attempt to restore homeostasis and promote cell survival. So some historical data that's important is that there is a special relationship between the integrated stress response and the pancreas developmentally, especially around the PERK arm. So PERK knockout mice best I can tell, the pancreas does nothing in utero except grow because it doesn't need to make digestive enzymes because you're being fed through the umbilical cord and mom controls your blood glucose. But as soon as the mouse or the baby is born, the pancreas has to go to work. And what happens in these mice is that within a few weeks of birth, all their pancreatic beta cells die and they get diabetes. Now, this looks very similar to a rare human syndrome called Wilcox-Rallison syndrome, in which humans inherit homozygous loss of function mutations in PERK. And these patients, within a few weeks of birth, all their pancreatic beta cells die, and they get infantile diabetes. Then they have other problems in the pancreas where the exocrine pancreas degenerates. PERK kinase inhibitors cause pancreatic beta cell loss and diabetes. And this is true even later on. If you delete PERK in the adult mouse, you see that quickly the mice develop hyperglycemia. So PERK's required for homeostasis of these cells as well. And beta cell specific ATF4, the transcription factor downstream of PERK, these mice have pancreatic beta cell dedifferentiation and exacerbated diabetes. So because of this, we have long been interested in this pathway and understanding what role this might have in tumors of pancreatic endocrine cells. And we've previously shown that ATF4, for example, is upregulated at the protein level and at the message level in human PanNETs, and that cell lines, pancreatic neuroendocrine cell lines, are sensitive to knockdown of ATF4 for their growth in culture. And we previously published that PERK pathway gets quickly upregulated in vivo when this is in the INS1 rat insulinoma pancreatic neuroendocrine model, in which these same cells can be grown on plastic, or you could take them and put them into a mouse in a xenograft, and you see that there is some PERK activation in a dish, but within 2 weeks, this pathway is very much turned on in vivo as the tumor struggles, probably because of hypoxia, low glucose, et cetera. So, moreover, if you knock out PERK in this model, the tumors have a very difficult time growing in vivo. You see that the tumors are much smaller. And if you inhibit PERK in vivo this is a GSK PERK inhibitor, kinase inhibitor, where we can do a pretty good job of inhibiting PERK activation, you see that in this model, the tumor is about 50% the size of the vehicle treated with just this single therapy. Now, going back to TORC1, recently it's been shown that TORC1 can translationally upregulate ATF4 in a mechanism that's independent of the integrated stress response, independent of EF2 alpha phosphorylation. And it does so, at least in some cells, through 4-EBP1 and de-repressing translation of ATF4 as well as increasing ATF4 mRNA stability. So we wanted to ask, does mTOR signaling support ATF4 expression in PanNETs? And then can the integrated stress response reactivate ATF4 in the setting of mTOR inhibitor resistance? So we see that in human cell lines, in this case of PanNET cells, BON1, QGP1, that very quickly, this is 24 hours after everolimus treatment, you get downregulation of ATF4. You do have downregulation, actually, interesting enough, to some extent of phosphorylation at EF2 alpha, but it's nowhere near as dramatic as the downregulation of ATF4. So most of this downregulation of ATF4 seems to be independent of the integrated stress response. Then trying to understand targets of ATF4 that might be relevant to PanNETs, we revisited published data using in the dependency map published data and mined it for targets that are both highly upregulated in QGP1 and that show when you knock them down, you have a big effect on cell viability. And one of the top targets that we mined out of that is SLC1A5, which is the predominant glutamine transporter in most cells. And also, it is known to be a target of ATF4. Importantly, I'll show you that in a second. And mTOR inhibition rapidly downregulates SLC1A5 expression in PanNET cells. You can see that here. It's slightly delayed, and it's after the downregulation of ATF4, but you can see at 72 hours it's very down. And forced expression of ATF4 promotes SLC1A5 expression. In this case, we're using HEX cells here. We're repeating this in the PanNET cell lines, but I suspect it's going to be similar. When you overexpress ATF4, you very quickly upregulate SLC1A5. And human PanNET cell lines are very sensitive. We basically confirm the data of the dependency map that if you use CRISPR to knock down SLC1A5 in QGP1 or BON1 cells and do colony forming assays, they do not grow well at all. And there's a reasonable inhibitor of SLC1A5 that's called V3902, and they show the IC50 of this is about, in most cell types for inhibiting the transporter, is about 10 micromolar, which is in the range that we're using it here. And the human PanNET cell lines show dose-dependent decrease in colony forming in the presence of the pharmacological inhibitor of this transporter. Likewise, these PanNET cells are very sensitive to glutamine deprivation in culture. So you can see that if we deplete glutamine from the media, you have a big impact on both QGP1 and BON1 cells. So we've also recently established, like others have, everolimus resistant versions of BON1 and QGP1. This is a log scale for the dose of everolimus, and you can get in the case of BON1, you can get a reasonable amount of resistance to everolimus in these cells, but it's especially true in the QGP1. They're even more resistant. But we've been using this as a way to start looking at everolimus resistance in this disease. And we see that both in the resistant versions of both of these cell lines, you see that you have marked upregulation of, I should say, I got an extra P in there, phospho-EF2 alpha and ATF4 in the resistant cell lines. And likewise, SLC1A5 is upregulated in the resistant versions of both these cell lines as well. And even though these cell lines, I just showed you a minute ago how resistant QGP1, the resistant version, is to everolimus, even though it's very resistant to everolimus, it is just as sensitive to inhibition of SLC1A5 as the parental cell line, suggesting that this is a relevant target, at least in the setting of resistance. So we're just at the beginning of this story, but I just want to summarize a few of the early observations. So TORC signaling clearly upregulates ATF4 and PanNETs. SLC1A5 is transcriptionally upregulated by ATF4, and it imports glutamine to help maintain metabolic demands in the cell. PanNETs can alternately reactivate ATF4 and SLC1A5 through another mechanism to resist mTOR inhibition, or in the presence at least of mTOR inhibition, which is depicted here. So when normally mTOR is leading to ATF4, turning ATF4 on, when you inhibit mTOR, it's clear that EF2 alpha phosphorylation goes up and ATF4 goes up. And this likely is suggesting that this is happening through the integrated stress response. And this seems to be at least one mechanism to help these cells resist. And hence, nodes in the integrated stress response may represent therapeutic targets in PanNETs. And so the ongoing studies we're doing now is really to test the role of the integrated stress response and this glutamine channel on PanNET growth and everolimus resistance in vivo, because we know oftentimes cells switch their metabolic state from in vitro to in vivo. Determine which arms of the integrated stress response are activated. We've already done a lot of work on PERK, but is that true in the setting of mTOR inhibition? And are there other arms of the pathway that are activated? Measure the mTOR and ATF4-driven metabolism, both glutamine and other metabolites in the PanNET cell lines, and then examine ISR activation in primary human PanNETs on a larger scale. These are the folks in the lab that did the work, especially Greg and Anjali. We have a lot of collaborators at University of Chicago. The cell lines came from Don's lab originally. Thank you so much to NETRF for the funding for this and other funding over the years, and I'm happy to take questions.
All right, that was a fantastic talk.
Thanks.
We'll take a few questions. I might start really quickly, Scott.
Yeah,
So,
appreciate it.
how do you think normal cells would react if you targeted SLC-A1?
Yeah, great question. I don't know. I mean, I guess it's like similar to many other things. The question is there a greater dependency? SLC1A5 in the dependency map, I'll just say that, does not, the other thing we screened out that I didn't talk about was common essential genes. So it's not a common essential gene, at least if they look across all the cell lines that were screened in the dependency map, it does not come up. So that, I should have mentioned that earlier, but that was an important thing that we screened out. So, DNA polymerase was not amongst our likely targets. But we need to understand that better, clearly. Yeah.
Okay, really good point. Thanks.
Yes. All right.
Emily?
Just a quick question. Really interesting. I was just wondering how sustained, like if you have sustained exposure to everolimus, for example, are these up or down regulations stable over time? Like have you carried out your experiments any farther than just a couple of days to see whether it's sustained?
Sure. So the everolimus resistant cells were, they were maintained at everolimus for months. So the expression of the upregulation of the integrated stress response certainly is maintained for at least months in that setting, or rises over those months. We haven't looked exactly at the time course, how kind of quickly it comes on, but importantly, what we see is that mTOR signaling is off still that in the setting of everolimus, and you're getting upregulations pathway, and we're trying to understand, how relevant is it for this, and then also just generally we're interested in this pathway as a general resistance mechanism. What we find here probably may not only be relevant for everolimus, but it may be relevant to other things that we do to target this cancer. Thank you.
Great, thank you. Just actually one question on the end there. Did you say what the mechanism of resistance was? Are you seeing mutations or you don't know?
No, we're doing all the sequencing on that as well. We have a number of other targets, but at least one of the things we're looking at this particular idea of do you upregulate these stress responses as a way to resist, but there's probably other things that are happening as well. Yeah.
All right, thanks, Chris. The next speaker, Scott. The next speaker will be Chris Harris, and they're from Rochester Medical Center, and he'll be presenting on ARID1A mutations and their function in metastatic neuroendocrine tumors.
I'm going to talk about real pancreatic cancers. Because I studied neuroendocrine tumors for like 10 years, but the last 3 I've been doing adenocarcinoma work for the most part. And we've identified a pathway that I think is also relevant to neuroendocrine tumors. I wanted to tell you about it. So pancreatic cancer is a highly lethal disease. It's the 10th most common disease, but it's the 3rd in total deaths. And mostly the problem is that it's really metastatic, where only 15% of the patients that come into the clinic can actually be resected. These are the lucky patients on the right. But you can see even within them, about 75% are going to have a very early recurrence. They only get treated by chemotherapy. What we're really looking for with the patients on the right is some way to treat them so that we can reduce the amount of this really fast metastasis between the time that they get diagnosed and get resected, because that's often 6-8 weeks. And during that time, there's a lot of new mets being built up. And I think we may have a drug that may be helpful here. We've been studying NETRF, the one that's in the box here. It's a secreted factor that is important in setting up the embryonic nervous system, okay? And after that, it gets shut down. So most cells don't express netrin. But you see it coming up again in a lot of cancers. So it's one of these fetal genes that wakes up. And it binds to several types of receptors. DCC we've heard talked about in ileal nets in the past because it's often lost. UNC5B is another one. That's the one that's in pancreatic cancer, both pancreatic neuroendocrine and pancreatic adenocarcinomas. So we'll talk mostly about that. Okay. We've done many screens for metastasis genes, neuroendocrine, adenocarcinomas, breast cancer, and we often come up with netrin being upregulated. This was a study that we did where we put in pancreatic adenocarcinoma into an orthotopic model, and then after 8 weeks, we cut out the Mets, and the Mets were all upregulated in netrin. This is a RipTag model that I set up, and Minah talked about this. When you put RipTag in BLAC6 or BLAC6AF1 or ABLAC6, you have an increasing amount of metastasis, you have an increasing amount of netrin. So we're always really interested in netrin. We could also go into clinical samples. These are adenocarcinomas. You can see IHCs showing more and more staining of netrin as the patients get more and more metastatic. Within patients with neuroendocrine tumors, you see de-repression of the netrin promoter in the metastatic tumors and not in the primaries. Okay, we knocked out netrin in mouse cell lines and orthotopically implanted them and watched them metastasize. And we can see that when we knock out netrin, there's less metastasis. I told you UNC5B is the only netrin receptor that's present. We've knocked out UNC5B in the same cell system, put it into animals, and they don't metastasize. And they're also very uninvasive when we do this. Okay. So we've also done this in genetically engineered mouse models. This is KPC. It's the major mouse model. It's like the RipTag mouse model of adenocarcinomas. And when we get rid of UNC5B, there's no metastasis. It completely goes away. And we've never seen anything like this before, okay? You don't usually get a single gene that's gonna do this. I think there's one other case of parathyroid-like hormone that does something similar, but this is a really strong effect. And we keep wanting to get funding so we can do this in RipTag mice, and we haven't done this yet. Okay, so we know that netrin binds to UNC5B and causes metastasis of adenocarcinomas. There is a drug, I don't work for NETRF Pharma, but I'm going to talk a lot about NETRF Pharma's drug. They made a monoclonal antibody that blocked the interaction between netrin and UNC5B, and it's this one. It's sometimes called MP137. And we've put this into mice these are splenic injections, and then we treat the mice with MP137, and you can see that they don't metastasize anymore. Well, you can see some, but it has a very strong block, okay? And it also helps the survival of the KPC mice when we treat them with this drug, so it's an interesting drug. Works very well in mice. These are neuroendocrine cells that we've looked at. You've heard about BON and QGP1 and CM. It's another one. CM's really mesenchymal, whereas BON and QGP1 are these very epithelial ones. These don't make METs very well. When we implant them into, I think we put these into the left ventricle, and the CM metastasizes much better. Invasiveness, CM is off the charts. BON and QGP1 don't invade very well. CM has netrin, BON and QGP1 don't. They all make UNC5B. When we treat with NP137, we can kill the CM cell, which is very resistant to anowekus when we're making it here. BON's not very resistant to anowekus, but the NP137 doesn't do anything to it. And we've done this again and again in many adenocarcinoma cell lines. And basically what happens is every time we have a mesenchymal cell, it is very resistant to anowekus and very sensitive to NP137. Whereas all the epithelial ones, shows strong anoecus, and the MP137 doesn't do anything. So this is the first drug that I know of that is preferentially toxic to mesenchymal cells. And drug companies have been looking for something like this because a lot of treatments, chemo and immunotherapy, work pretty well on epithelial cells and don't touch the mesenchymal cells. So they've been looking for something that they can combine with these treatments in order to increase the efficacy of the single treatments. So we've worked out the anoecus assay or mechanism. I don't want to talk about this right now, it's too complicated. That was a lot of work, though. We also find that netrin UNC5B signaling activates the epithelial mesenchymal transition. Most adenocarcinoma cell lines are very epithelial. This is an exception, A1925. It's very mesenchymal, doesn't make much coherent. But when we knock out UNC5B, it reverses. It goes back to an epithelial state, okay? When we look at knockouts, EMT is the -- alteration by enricher analysis of RNA-seq data. Netris Pharma has a phase 1 study that just got published in Nature. And what they see that's really interesting is that, and these are endometrial cancer patients, and they were biopsied before treatment with MP137, and then 4 weeks later they were biopsied again, and it was analyzed, and you could see that the EMT score drops quite a bit when they get treated, when these patients get treated with MP137. So the MP137 is having a good effect in vivo in the clinic on different tumor types. Okay. We presented our pancreatic adenocarcinoma data at AACR Pancreatic Cancer last year, and immediately 3 clinical trials got announced, one of which is ours, which is about to start next month, but 2 months from now. LAPNET1, which is done by Netris, is in France. It's already recruiting patients, already treating patients. And SWOG talked to us a lot, and they decided to make MP137 one of the arms to a big FOLFIRINOX plus X trial that they're doing. So they're doing like 80 patients. We're doing 40. And Netris is doing 50. So out of all this, what I'm really looking for is we're going to biopsy before they get treated. They'll get 2 cycles. They'll get biopsied again when we surgically resect them. These will all be resectable patients. And then we'll look to see what happens with them, okay? We'll also do, follow-ups and look at their effects on metastasis. So now I told you that netrin is shut down in normal cells, and we've been investigating what turns it on. And what we found is that ARID1A mutations, which are found in about 20% of all cancers, including like lung nets, will make pancreatic adenocarcinomas become very mesenchymal. And at the same time, they will turn on netrin. It's the number 56 change at the RNA-seq level. The protein goes up, and we can do single knockouts in different cell lines, like a neuroendocrine or an adenocarcinoma, and we can turn on netrin. So ARID1A is alone sufficient for this. In ARID1A mice, the knockout itself causes many changes. 2,000 genes get changed, but the number 1 is EMT, and these are the number 2, 3, 4, 5. And when we knock out UNC5B, look at what gets re-established. The same ones in the same order, showing that of all these 2,000 genes that get altered by ARID1A knockout, UNC5B seems to be one of the most important. So it does provocative things in the context of ARID1A knockouts. You may not know this, but ARID1A is the most mesenchymal or metastatic associated gene in pancreatic neuroendocrine tumors. If a patient comes in, it's an uncommon mutation, but if a patient comes in with an ARID1A mutation, almost always it's a metastasis, okay? DAX and Atrix also highly associate with metastasis, but not like ARID1A. Okay, and this came from Otter Singh's report 2018. Patients with ARID1A mutations don't do well with pancreatic neuroendocrine tumors. Okay, so this is ARID1A. It's part of the CBAF, which is a SWI-SNF complex. It's this one right here. We also find that netrin-1 becomes de-repressed in patients that have Atrix and DAX mutations. These are pancreatic neuroendocrine tumors. So these are the wild-type patients, and these are the Atrix-DAX mutants. So you can see that Atrix-DAX is another way that this thing can be woken up, which was interesting. I already showed you this, that if we knock out ARID and QGP1, we can activate netrin-1. We just got Atrix knockout cells from Chris Efi, and we can see that Atrix gets turned on when it's knocked out versus the wild type QGP1. This is the Atrix knockout. So it looks like Atrix is enough to de-repress. And that those cell lines, remember I told you about CM is the only one that expresses netrin? We sequenced, and CM is the only one that has an ARID1A mutation, right? BON and QGP1 don't have one. So this is another example of ARID1A associated with upregulation of netrin. So I think that netrin is really important in the metastatic pancreatic neuroendocrine tumors, almost all of which have Atrix, DAX, or ARID1A mutations. And I think that it's very possible that, that patients with these sorts of mutations could be treated with NP137 the same way we're trying to treat pancreatic adenocarcinoma patients, right? And finally, we did this just last week. We looked at deposition because we see that ARID1A can affect deposition of a repressive mark, histone H3K27 trimethyl, and you can see that it drops precipitously on the netrin promoter when ARID's knocked out. But look what comes up. There's this activating mark, H3.3, the same H3.3 that's altered that's dysregulated by Atrix and DAX mutations. That may be why Atrix and DAX mutations also can turn on netrin, but that's an experiment we haven't done yet because your cells are growing too slowly. Okay, so to summarize, in both types of pancreatic tumors, netrin de-repression stimulates metastasis by decreasing anoecus and promoting EMT. We can block this with NP137, and we're putting this in clinic right now. And netrin de-repression correlates with the H3.3 deposition. So we're looking highly at that right now, especially in adenocarcinomas where H3.3 has never really been looked at before. And we're starting to see that it's linked to some other interesting genes as well. So, and this is the crew at Rochester. We just lost them. Oh, we're always going to lose them. Okay, Darren Carpizo is the head of the lab. He's the chief of surgical oncology at Rochester. And I also worked with Igor at Satcharov, who's at Fox Chase, and he's been great. And Patrick Malin is our contact at NETRF Pharma. He's been working on netrin and UNC5B for 25 years. So he's a great resource. So thank you.
We're a little bit behind, but maybe we have time for a question if there is one.
Very quickly. First, Chris, great presentation. I want to thank you for making the RT2F1 model. It's been a fantastic model for my work as well. Just a statement, we have cell lines from the RT2F1. I'm happy to send them to you to see if this NTN1 and the antibody is a target, and also happy to collaborate with the organoids if you want. But what we see as well from characterizing your model is that a lot of these classic neuronal genes are upregulated, and NTN1 is one of them. So great work. I think it will be a great target that we should explore for NETs.
Okay.
Just a statement. Thank you.
Thank you.
Up here. So I'm going to ask Chris to stay up here and then ask the other speakers who are in this group to come on up before our last speaker goes, and then we'll be all ready. Oh no, if you can come up now, you can get situated. And then meanwhile, our last speaker is Panagiota Kafasla. Oh, I got it right. And she is going to tell us about altered splicing programs in pancreatic nets affecting the function of neuroendocrine cells.
So, hi everybody. Yes, okay, thank you. So I would firstly like to thank NETRF, not only for giving me the opportunity to talk today and present our results, but also for giving me the opportunity to start this project, actually. And it's getting more and more an exciting project, as you will see. So I'm going to be talking to you today about the work we are doing on pancreatic neuroendocrine tumors and how splicing changes in these tumors. So I would like to remind you that alternative splicing is an RNA, a nuclear RNA processing event that contributes to the high diversity of eukaryotic gene expression. And how does it do that? It does it by promoting, I need to get used to it. The production from a single gene of many different mRNA isoforms with the inclusion or the skipping of exons or of certain sequences of the pre-mRNA. So by getting different mRNA isoforms, one can understand that we get a different translation of different protein isoforms that may differ in domains that are included or not. So these protein isoforms might have different stability, different localization, or function generally. So it's now known that alternative splicing is deregulated in cancer. That's a characteristic of cancer. And in tumors, we have more than 30% of more events, more alternative splicing events taking place compared to normal tissues. And it's also known that tumors contain many splicing isoforms that are not detected in tissues from healthy individuals. So one can understand that because of that, there has been some extensive research in targeting alternative splicing deregulation in disease for therapy development and in cancers. And some approaches, I'm just going to mention a couple, is either the generation of small molecule inhibitors of splicing factors, which many of them are in clinical trials. But also there is this elegant way of using the antisense oligonucleotides, not only to downregulate splicing factors, but also to change the outcome of the splicing of a certain pre-mRNA specifically. So when we started with this project, there was not much known on pancreatic neuroendocrine tumors and deregulation of alternative splicing. I'm not going to say more about these cancers, of course, you know about them. But our questions starting were the following. We wanted to see whether alternative splicing can be also a source of therapeutic targets in PNETs, in this type of nets, and whether we could identify a splicing factor signature that is altered in PNETs, and whether we could identify alternative splicing-derived isoforms that might be upregulated or downregulated in PNETs during disease development. So to do that, we started by bioinformatics analysis, and we analyzed a large number of publicly available RNA data sets. As you see here, we had a large number of samples, 62 PNET samples, 50 of them from non-functional tumors, and 67 normal samples, 39 human normal islets in this set. So what we did, we compared PNET to normal samples, not only on the expression levels of splicing factors, but also on the levels of alternative splicing-derived mRNA isoforms. So for the first part, regarding gene expression analysis of splicing factors, of course, we could detect a large number of splicing factors being deregulated. And with quite stringent thresholds, we could detect 26 splicing factors being overexpressed significantly in PNETs compared to normal human islets. And 6 of them, interestingly, are splicing factors that are known as neuronal factors. Most of them do not really express at all in other tissues. we started looking for some differences between non-functional and functional tumors. There are some differences, but we need more samples to be more confident about our results. So we also wanted to identify, if there was such a possibility, a splicing factor signature that might be specific for PanNETs. To do that, we compared the expression levels of the splicing factors that are deregulated in PanNETs to their expression levels in other cancer types from the TCGA database. And we did get a small subset of splicing factors that seem to be deregulated more specifically in PanNETs and compared to these other types. And this group is actually the neuronal splicing factors that I said earlier. So when we compared the expression levels of these factors to their expression levels in other nets, I have to say that we saw a similar deregulation. A couple of splicing factors might be related to more aggressive PanNETs, but still we need more samples here to be able to have conclusive results. Going to the analysis of the alternative splicing-derived mRNA isoforms, what I have here is just a PCA plot that has been generated based on the expression levels of the isoforms. And one can easily see that alternative splicing can differentiate PanNET samples from normal islet samples or other single-cell pancreatic populations. So we focused our analysis first on the alternative splicing isoforms that are being generated due to differential inclusion or skipping of exons, which is the largest number of events that we detect. And to go further, we try to see, okay, we have all these alternative splicing events identified. Can we correlate the events with some of the splicing factors specifically? Can we predict which splicing factor actually regulates these events? So we looked for many splicing factors, but we would like to find a regulatory splicing factor rather than a core splicing factor, which would be important for the whole splicing that takes place in a cell. So doing these correlation studies, we identified SRRM3, which is a neuronal splicing factor overexpressed in PanNET samples, as a factor that possibly regulates the majority of the alternative splicing events that are deregulated in PanNETs. And when we looked for the functional effect these deregulated alternative splicing events might have, we performed gene ontology enrichment analysis, of course, for the genes that are subjected to alternative splicing changes. And we saw, you cannot probably see it here, but you can definitely see it here. To our surprise, they were, all of them, most of them, important for vesicle organization, transsynaptic signaling, Golgi vesicle transport, endocytosis, and generally functions of the neuroendocrine cells that are important for hormone secretion, but also for receptor endocytosis and so on. So after that, we wanted to continue with some wet lab experiments, and these are the results that I will present. So for our wet lab experiments, we used human PanNET cell lines, BON1, QGP1, and NT3, but also we used the RIPO-ANTAC2 mouse model, which people have shown before. We used, though, 3 different time points of this model. So somehow at the beginning, at the early stages, when we have islet hyperplasia, and at the late stages of the model. I have to say that from the very beginning, we stopped using BON1 cells because the alternative splicing pattern in these cells is completely different to the ones that we saw from PanNETs, from bioinformatic analysis. And using these cells, we first of all perform validations. So I'm just going to show you our results for SRRM3, for example, as an example. We validated by qPCR that the SRM3 mRNA levels are increased during the disease course in this mouse model compared to the wild-type mice. And we validated the expression levels changes in the expression levels of the rest of the splicing factors. And we also validated the changes in alternative splicing isoform levels. I have put here just one example, and that I will try to explain. We use RT-PCR and gel electrophoresis in the lab. With specific primers, we can detect the inclusion, the mRNA that has included the alternative exon, but also the mRNA that is the product of the skipping. So one can see that at the last 2 time points of the model, of the mouse model that we are using, we have the inclusion band appearing compared to the wild-type mice. And this result here has to do with is the result from an mRNA that codes for a protein that functions in ER to Golgi vesicle-mediated transport. And in fact, the inclusion or not of this exon alters the ER lumen domain of this protein. So we also perform validations on the dependency of these events, alternative splicing events on SRM3. We downregulated SRM3 using a specific SH, and we saw that when we downregulate SRM3, we have decreased inclusion of this alternative exon. So indeed, the regulation of these events takes place through SRM3. Following that, we needed to check whether SRM3 has actually an effect on the tumor characteristics of PanNET. And for that, we used the QGB1 cells treated with shRNA specific for SRM3 or not treated, and we performed cell viability assays. The better the downregulation was, the worse it was for the cell's viability. We also performed colony formation assays, reduction in colony number and area when we had downregulated SRM3. And of course, the morphology of the cells changed also upon downregulation of SRM3. We had not really that large spheres being formed. And when we use these cells in xenografts, we could detect a reduction of tumor size. Of course, as I said, the majority of the splicing events that we found to be upregulated in PanNETs compared to normal islets affected the production of protein isoforms that participate in many steps in endocytosis, hormone secretion, vesicle-mediated trafficking in the cell. So we have started doing experiments on the importance of SRM3 on these pathways. And I just have some first results that show that upon SRM3 downregulation, the intracellular calcium levels upon glucose induction are reduced compared to the wild type, the non-treated cells, if you want, and that the cells upon SRM3 downregulation secrete less insulin. We also wanted to check what's happening in the cell in the actual vesicle trafficking machinery, and we have started doing experiments, and we just saw that upon SRM3 downregulation, we see a significant difference at the Golgi network, I could say it, as you see here. So our next steps first of all are to work a bit more on the mechanism, what happens, how SRM3 exactly downregulates the regulates the vesicle-mediated trafficking. But we also want to, if possible, identify alternative splicing changes that are responsible for the cancer characteristics of PanNETs and for the changes in vesicle-mediated trafficking. And of course, we're going to study more, as I said, the implication, the involvement of the SRM3 in these properties. Another thing, another part of the project is to compare, to get more samples and expand our bioinformatic analysis to be able to compare functional versus non-functional tumors or ADM mutant versus non-mutant tumors. And this is something we will be doing in collaboration with, using different mouse models in collaborations with Amanda, with Professor Bibbs that I met here last year. So I'm very happy to see them again this year. And we have also started a collaboration very, very recently with Professor Kantsas in Greece, in a hospital. He is able of to give us RNA samples from patients, and doing a literature research, I have seen that most of these mRNA or protein isoforms that are subjected to alternative splicing can be actually detected in blood samples, in plasma or exosomes from blood from patients. So we might find some markers there, or some material from patients. And with this, I would like to finish by thanking, first of all, my people in the lab, my colleagues, NETRF, other funding agencies, and this is where we do this work in Greece. Thank you very much for your attention. Thank you.
Thank you very much. We could take one question and then we'll open it up for a short discussion before our break.
I have a question and a comment. MEN1 is right next to NEAT1. I don't know if you know that. Mm-hmm. They're on the same chromosome. And 75% of patients with PanNETs lose both. And that's going to change the splicing. For instance, do you see paraspeckles in pancreatic neuroendocrine tumors? Have you looked and can compare that? Because if you lose the NEAT1, you're going to lose the paraspeckles, and splicing is going to. You might want to look at that. And it's easy to model that in mice. We actually have NEAT1 flocks in the next lab, and I've thought about making that, but
that's a very good thing. That's a very good point. And in the same location, there is also, because I was checking it, SF3B1, which is a very major splicing factor, is very close to MEN1 as well there. Yeah. So that would make sense to have a look at all
so I think just by the function of MEN1 being an important driver, it may be linked to all these splicing. The other thing is you should make RipTag ABLAC6F1s.
Yeah, I know.
Right? Because that's going to be the non-functioning version of the BLAC6. Yes. All right. You can forget everybody else's mouse, but that's the one you want. So.
Okay. Thanks. Thank you very much.
All right. Thanks. So if you can just have a seat. So we'd like to open it up, but we are very tight because I guess we're not good moderators, and we've gotten to the end of our time almost, or we are at the end of our time. But if we could just take 5 minutes and ask anyone if they want to probe any more questions with any of the speakers, or if the speakers actually have questions for each other, because you're up here and you don't have the chance to do that.
Okay, actually, I had one quick question, or maybe. So this is kind of for Minah. I think one of the themes to me that emerged from the different talks was autocrine signaling and autocrine activation. So I think we had ATF4 activating amino acid uptake and then driving its own growth. TMEM127 upregulating RET, causing autocrine growth. And then netrin going to UBC5. So we're just seeing this kind of cell autocrine loop. And Minah, you didn't tie in Anschutz to an autocrine loop. And I wonder whether or not that's something you looked at, or whether that's something you would think about in your model. For you. So the short version is there an Anschutz autocrine signaling pathway that could exist to be driving growth? Because you focused on an endothelial interaction.
No, I mean, the question is the autocrine effect of angiopoietin in endothelial cell itself. So we do usually, when angiopoietin 2 suppresses Tie2 signaling pathway, We didn't look at further the downstream pathway, but what I studied before is angiopoietin-2 can suppress Tie2. And as a downstream molecule, we studied like a FOXO1 transcription factor, which can also regulate angiopoietin-2 expression, endogenous expression itself in endothelial cells as like a positive feedback loop. But later than that, we didn't really focus on this autocrine system.
I'll ask a question of Minah because it's the same problem that I'm struggling with, and that's that you're adding your antibody after the metastatic cells have already colonized. And what we want for patients is something we can add when the patient presents with a metastasis. Mm-hmm. And have you ever thought about treating them after a really good metastasis formed? And with that, I know the primary is growing really fast. I know this model, but we want something that will eliminate that. And I don't actually know how to model it. So have you had any thoughts on that?
So we are wondering about when we use this RT2AJ AB6F4 mice, we are also wondering about the effect of angiopoietin inhibition at different time point of metastasis. So when we treated this anti-angiopoietin-2 at 15 week, as you know, this 15 week we see kind of microscopic metastasis. It's quite very early stage of metastasis. And we did like 18 week is a kind of later stage of metastasis. And also we did like a long-term treatment, like 8 weeks until 23 weeks, very late stage of metastasis. And luckily we found very consistent data regardless of the early or later stage of metastasis, which is very interesting. Because when we block anti-angiopoietin-2 at different time points, like at 15 and 18 week, we could see the differential effect in primary tumors. Because we only saw this anti-tumor effect in the later stage when we block angiopoietin-2 for primary tumors. But for liver metastasis, we could see kind of consistent effect, which was good.
I'm a big fan of that model. Mostly because it's cabozantinib, it looks like it's going to get approved. It came out of riptag mice, and everolimus came out of riptag mice, and sunitinib came out of riptag mice. If you can get a drug to work in riptag mice, I think you have a really good chance of getting it clinically approved. It's also, that mouse has also shown like 3 drugs that didn't work in riptag mice that went into clinic and failed. And I think it's a really good mouse to do clinical studies on because it's predictive both ways. I wish we had something like that in adenocarcinoma. We don't have that. So I really encourage people to use the riptag. NETRF as sketchy on funding riptag work. It's not their fault. I think the people that read the grants often will list a weakness of this grant is that it's using riptag mice, and that shouldn't be. That should be a strength of a grant. And I hope going forward that they'll think about doing things this way because it's a really good clinically proven mouse that can lead to patient treatments.
Great, good comment. I was lucky to have funding.
All right. Well, thank you all. If we could give a hand to all of the speakers. And please.
So this will be our last section for the day of our NET pathways and targets. This is going to feature a few talks on gastrointestinal neuroendocrine tumors. My name is Jess Maxwell. I'm from MD Anderson. Co-chairing with me today is Dr. Ramesh Shivdasani. So appreciate you guys hanging tough. I think this is going to be a great session with some really interesting topics to be discussed and some that I hope we can have some really great discussion at the end. So I'd like to first welcome Dr. Pobin Ear, who's from the University of Iowa, to discuss investigating serotonin metabolism in neuroendocrine tumors.
Thank you. So first of all, I would like to thank the organizer for giving us this platform to present our work on mainly the regulation and metabolism of a neuroendocrine cancer here. So in Iowa, our team of clinicians see a lot of patients with small bowel neuroendocrine tumor, up to over 50% of our patient population actually have SBNET. And this is why we want to really understand the tumor metabolism of this cancer and also find ways to help improve the treatment. Because as you may all know, there this class of neuroendocrine tumors that are really highly drug resistant. And it seems like, the approved therapies out there just don't work well. So lately we have some preliminary data or that's been going on for a few years now that we've compilated. So we did a lot of drug screens with the patient tumor spheroid. And when we put our data together, what we realized is that the small bowel neuroendocrine tumor spheroid, they are highly drug resistant, similarly to what our clinical colleagues see with the small bowel neuroendocrine tumor patients. As you can see here, the summary of this is that's the small bowel neuroendocrine tumor spheroid. They're only sensitive to 21 drugs of the 175 that we tested, in comparison to PanNET spheroid, where they were sensitive to more drugs, and the neuroendocrine carcinoma, they were actually sensitive to many more drugs. One feature, of course, of small bowel neuroendocrine tumor is that they express and they produce a lot of serotonin, and these are quantification by our pathologist, Dr. Bellisi, and as you can see, up to 80-90% of these small bowel neuroendocrine tumor actually stains positive for serotonin. And so we think that it's really important to study the serotonin metabolism in small bowel neuroendocrine tumor. Just for a recap, the serotonin that is produced in small bowel neuroendocrine tumors, they are being produced by the TPH1 gene. So this is tryptophan hydroxylase 1. There is the FDA-approved drug, telitristat ethyl, that is approved for SBNET patient with carcinoid syndrome, and the active form of the drug, which is telitristat, actually directly inhibit the enzyme TPH1. So we characterize the expression of TPH1 in small bowel neuroendocrine tumor relative to the adjacent normal tissue, and we see a really high level of TPH1 in SBNET, both primary and metastatic tumors. When we do staining, we see that they are really localized in the neuroendocrine tumor cells, not around the fibroblasts, for example. It's really within the tumor nest here. And lately, Dr. Bellisi have developed an IHC protocol for staining for TPH1 as well, and we see that it actually recapitulates what we see by immunofluorescence. So studying serotonin metabolism is actually really hard, and we first start to just try to inhibit the pathway using cell culture models, and then we didn't see much happening using the inhibitor, so that was kind of discouraging. We even spent like months developing these cell lines where we knocked down TPH1, and we got these stable clones and all that, and we spent months on them, and then we look at the proliferation rate, and there was nothing. So it was really sad. So we thought, okay, well, maybe we should give up, but let's just try one more experiment. Let's just put it in the mouse and see what happens. And that's when we put it in the mouse, and we saw this really dramatic phenotype. So the TPH1 knockdown, the tumors, actually, they grow super slowly, as you can see here with the blue lines. So that was the genetic knockdown model. So then we want to see, so if we use the inhibitor of TPH1, can we recapitulate the phenotype? And we were lucky that actually we can actually see this as well when we use the TPH1 inhibitor telotristat ethyl. We can also reduce tumor growth. And we saw this with the subcutaneous model, but we also use a liver metastases model where we inject the tumor cells in the spleen and then they migrate to the liver. And then we do luminescence imaging to quantify the tumor, and we actually saw this. And this was at this time, Dr. Metz, David Metz group also found this in the clinical trials. So that was really encouraging, and it really motivates us to continue further. So we took our tumors and then we look at them more at the molecular level, and we looked at the gene expression. And what we noticed is that TE-treated tumors, they actually express less some of these drug metabolizing enzyme and also these drug pumps to pump out like the anti-cancer drug. And so we thought that, well, this is great. So it means like maybe we can give them the TKI or other drugs that is approved for neuroendocrine tumors, and maybe they will respond better. And this is what we saw when we combine TE with sunitinib, we actually get a better reduction in tumor growth. We also measured the serotonin level in these tumors, and we saw that actually the combination therapy improved the serotonin. Actually, the tumors has really a lower serotonin levels as well. So to look further, we want to see what's happening in the tumors that we inhibit TPH1, and we noticed that, and we stained for angiogenesis marker, just because like a few pictures ago, I showed you that the tumors actually, if you just look at the tumors, they look less vascularized. They were not as reddish when we harvest them. So that's why we looked into the endothelial cells that were in the tumor section, and we noticed that they were actually less endothelial cells stained in green here for the control group compared to the TPH1 knockdown. There were also a reduction in the VEGF A, B, and C and at the gene expression level. And then when we performed this conditioned media experiment where we took conditioned media from either wild type tumor cells or the one that has a TPH1 knockdown, we saw that actually they can impair the knockdown. The one that has less serotonin, they actually impair growth and connectivity of the endothelial cells. And this phenotype is actually, you can actually rescue it by pipetting back serotonin into that conditioned media. So in my lab, we wanted to dig a little bit deeper, but of course, again, studying serotonin metabolism is quite hard, and I'm a biochemist by training, and I find it hard. So this is why we decided to develop other tools that we can use for studying serotonin metabolism. So, and luckily, recently there's this green fluorescent sensor for serotonin that was developed by the Tien lab, and we thought, well, it would be nice to actually adapt it to become a luminescent sensor, because the thing is, when you want to study in vivo imaging and all that, green fluorescent sensors are not ideal because our body has a lot of metabolites that fluoresce in that range. So that's why we adapted the sensor to become a luminescent sensor. So now this one, it can produce light, and then we can quantify the signal that way. So initially, we developed this in yeast because it's easy to do the cloning, and we actually got these results within a few weeks. So that's just how fast that this can be done. But of course, once we found the right sensor that works, then we adapted to the neuroendocrine cancer cells. So then we spent a few more months generating these bond cells that has a serotonin sensor, and we see that it's working. And then we wanted to see, so when does the serotonin level changes in the cancer cells? So we started with testing with many growth factor. We tested them at different concentration, at different duration, and then we didn't see much. So that was another little disappointment there. But then we thought, okay, well, maybe we're doing it the wrong way. Let's take away the growth factor and see what happens. So then that's when we saw drastic changes in serotonin level. So when we start removing the growth factor, we see a significantly increase in serotonin levels in the cells. And we looked at TPH1 expression. It actually didn't change much, but actually the phosphorylated form of TPH1 changed dramatically. So we kind of titrate this and to see if we can get a dose response effect. And so by decreasing different concentration of the serum, and we can see that we can actually get this nice escalation response. We look at how long it takes, and we realize that this is really a dynamic system, because within half an hour, you actually get increase in serotonin level to the max. So biologically, we want to see when do serotonin metabolism change in the tumor cells. Another condition is when they transition from really the adherent state, so they're growing in the bottom, stick to the plastic, and then they transition into the suspension state. And this is kind of mimicking tumor metastases, a simplified version. And we see that in this model, we also get really high expression of serotonin expression. So this was actually confirmed. So that the panel A and B, that was our sensor, and then we confirm it by using mass spectrometry. We also confirm it by immunofluorescence. So overall, this is our summary. We think TPH1, well, it's really the key regulator of serotonin metabolism in neuroendocrine cancer. It's very important for tumor growth in vivo, although in vitro, as I've shown, it doesn't do much, because I think, in vitro, we feed the cells, we give them excess amount of nutrient in the Petri dish, whereas in vivo, well, it depends on the nutrient that they get. It depends on the vasculature of the tumors. And so this is, in summary, and we've developed this sensor that allowed us to dissect this mechanism further. And so in summary, we think that serotonin metabolism increase when the tumor is growing rapidly, and so the tumor cells are competing for nutrients, so that's why they need more blood vessels to bring in more nutrients, and also when they metastasize. So with this, thank you very much. I was a recipient of the NANET's NETRF award 2 years ago, and now our work is being funded by the NETRF pilot grant, so thank you for making this possible. And also I would like to invite people in this room to join us for the NET Models meeting next year. So Talia Dalton and I, we will be co-leading the ex vivo neuroendocrine tumor models, and Dawn and others are co-leading the animal models and all that. And our goal is to really put together kind of like a review paper summarizing the resource and also the limitation and the advantages of these techniques. Thank you.
We have time for a couple of questions.
I get one. I was wondering, in your TPH1 knockout model, when you planted it in the mouse, you didn't mention the fibroblasts and the stroma that the serotonin is inducing in the primary tumor and in the heart valve and many places. Did you see any differences in the amount of matrix deposition?
Well, we clearly see the amount of the vasculature. In terms of the fibroblasts, I think they are more fibrotic, but we just haven't quantified them. But I guess that would be the next, it would be the really, it would be the to look at the fibrosis and the cancer-associated fibroblasts as the next phase of this project. Thank you.
Ion cells, when you grow non-adherent, they undergo analyses. And also you see probably if you're TPH1, which is what I'm thinking of range.
Yeah,
It's possible that your inhibitor would actually keep them alive when they're floating, which may not be what you want.
I think the experiment was done really like within a few minutes after you can put them in as well. So I don't think there's time for anuicus yet. And also what we saw was like these bond cells. I know in some cancer cells they actually undergo anuicus and they die, but the bond cells actually they can be growing fine as spheroids. So they clump together and they form these tumor spheroids, and they're kind of happily grown in their.
They also show a lot of anuicus. They're not that happy.
They're not.
There's some happy ones, but there's some, and they could be through the iron loss being produced by the TPH1. So I just worry about the drug promoting passes because it allows them to survive.
Oh, you're worried about Yeah. So, I mean, I think clinically, I don't think, teletristat ethyl is not solving the problem, right? I think many clinicians here can confirm that, the tumors are still growing. But I think there might be room for example, combining teletristat ethyl in combination with TKI, and that I think there are some additive or synergistic effect on angiogenesis pathway, but yeah, by itself, you're right. Yeah, you're right. Thank you.
Okay, the next speaker in this session is Maria Martins from University College of London, who will talk about mesenteric fibrosis in small intestinal neuroendocrine tumors.
Hello, everyone. I'm a PhD student at UCL, and I'll be talking about my group's work on mesenteric fibrosis in SINETs. So starting with a bit of background, as you know, the incidence of NETs has been rising, and if we see here for the small intestine in this black line, we see that is one of the sites with the highest incidence. And besides that, these patients are often diagnosed at a late stage, and over 50% of them present with metastatic disease at the time of diagnosis. So we are specifically looking at mesenteric fibrosis, or MF, which is a common complication in SINETs affecting up to 50% of patients. And this occurs when there's a fibrotic reaction around the metastases in the mesentery, which leads to the formation of a larger mass, which we can see here on the CT image. And this can then lead to other complications such as intestinal obstruction, edema, or ischemia. And looking at this graph, we can also see that we have patients without mesenteric fibrosis at the top, and in gray, patients with mesenteric fibrosis showing a significantly shorter overall survival. One of the main problems also with MF is the lack of understanding of the pathophysiology, which then limits the development of new treatments. And so our group is focused on elucidating further on that. So as we have presented here last year at the NETRF, our project is a collaboration between two ENET centers of excellence funded by the NETRF. So between Royal Free Hospital and the Erasmus MC Center. And we are focused on three main aims, which includes looking at factors and pathways implicated in the carcinogenesis and fibrogenesis by looking at patient databases, in vitro models, and also analyzing patient tissue. We are also looking at biomarkers for diagnosis and prediction of the clinical risk of MF using transcriptomic and proteomic data. And we are also developing a radiomics model. So today I'll be talking about some of the work that we have so far at the Royal Free site. So we have performed RNA sequencing and epigenetic analysis on a cohort of 45 SNF patients. We had normal intestine, primary tumor, and mesenteric mass tissues for these patients, and they had also been classified into 4 groups of fibrosis severity, which include none for no fibrosis and minimal, mild, and severe, which were done using histological, surgical, and radiological assessments. And then we extracted the RNA and DNA from fresh frozen tissue that had been collected, and we performed RNA sequencing using Illumina Novasec and DNA methylation analysis using the EPIC microarrays, and analyzed both the transcriptomic and epigenetic data, and also integrated this data in order to have a better overview of the profile of the disease. So this analysis is still ongoing, but today I'll be showing some of the preliminary results that we have. So in total, we had 25 comparisons that would be between the different tissue types and the different groups of fibrosis severity. We decided to first focus on these 3, which include the comparison between the primary tumor and the mesenteric mass in each of the fibrosis groups. On the top row, we have the volcano plots for the methylation data, and on the bottom for the transcriptomic data. And looking at this graph on the top left, we see that there weren't a lot of hits for the minimal fibrosis group, so we decided to focus on the other 2 comparisons. So then we selected only the genes that were significantly altered, both in the DNA methylation and in the epigenetic data for each of these comparisons. And we saw that there was an overlap of 32 genes, and that 9 of these 32 genes were already described in literature as being fibrosis associated, which would be this list of genes, and that we'll be looking further into in the context of MF. So another side of our project is the development of a 3D model in order to look at the interaction between cancer-associated fibroblasts and cancer cells. So this model in general is based on the idea of having human intestine that can be decellularized into a scaffold and then lyophilized into an ECM powder that can be jellified together with cells to obtain a native matrix where the cells can grow in. And so if we go into a bit more detail into this, we have, for the decellularization, we are collecting healthy intestine from our SNET patients that undergo surgery. We are also trying to collect the mesenteric mass, so that would be the fibrotic tissue, but this one is a bit harder to collect. So we freeze it first at -80 degrees and then thaw at 37, and then the tissue goes through cycles of washes in sodium deoxycholate, PBS, and water, and then we also take samples for quality control. We are also collecting primary fibroblasts from SNET patients, and we are currently collecting from 4 sites, which include the normal intestine, the normal mesentery, the primary tumor, and the mesenteric mass. So these are washed in antibiotics first before being then digested, and then we can culture the cells. And finally, the idea is once we have the ECM powder, we can make a solution and gellify it specifically with our primary calves that we isolate from the patients, and with GOT1, which is the cancer cell line that we are using to obtain our gels. So now we have been focusing on looking at cell viability of our cells in monocultures and co-cultures in these gels, and next we'll be looking further into the interactions between these 2 cell types. So here are some representative images of native tissue before decellularization and after decellularization. So these are H&E staining, so we show a clear distribution of the cells in the native tissue, while for the decellularized tissue, we only have the remaining matrix with no cellular content left. And here we have serious red staining, which stains in red the collagen fibers, and in yellow we have the cellular content. So we have a clear distinction here in the native tissue between these 2, while for the decellularized one, we only have the remaining collagen stain in the matrix. So then we cultured our gels. These are cultured for 14 days using, to start with, 500,000 GOT1 cells and 200,000 fibroblasts. And we performed Presto Blue, which is a cell viability assay at day 7 and day 14. And looking at the graph, we can see that for the calf monoculture, there wasn't a significant change. However, for the GOT1 monoculture and the co-cultures, there was a significant increase from day 7 to day 14. And here we have the H&E stainings for these gels. On the left-hand side, we have the calf monocultures that show a distribution of the cells throughout the gel, but there wasn't any significant change in terms of cell numbers from day 7 to day 14, which is in line with what we saw in the Presto Blue assays. While for the GOT1 monocultures and the co-cultures, there's a clear increase both in the number of the clusters and in their size from day 7 to day 14. So in conclusion, we've seen that the mesenteric mass has a significantly altered methylation profile that then leads to transcriptomic changes in the tissue of our SNET patients with MF. And we have also shown that the GOT1 and the calves have good viability in our 3D model using human intestine ECM. And these are the first steps into creating a 3D model that can be used to study the interactions between these two cell types using a native matrix. And then, for the next steps, we will be further looking at the epigenetic and transcriptomic data. We will also be further investigating the crosstalk between these two cell types, the cancer cells and the calves, in our 3D model. And we will also be selecting candidate genes to take further into evaluating them in vitro. So I would like to thank the NETRF for funding this project and giving us the opportunity to present the data here today. I'd also like to thank our patients and our team at Royal Free and UCL, as well as our collaborators at Erasmus MC and the University of Exeter.
Questions for Maria?
Very nice work. Maybe I've missed this. So you decellularize the sample you get from a patient?
Yes.
I wonder if, so this patient is a patient with neuroendocrine tumors?
Yes.
So I wonder whether you've tried to isolate also the primary tumor and just put it in and create a new model now, like a kind of the organoids, but now you're going to have the ECM, decellularized tissue, and then putting the cells you're getting from the patient. I think that would be very interesting.
Yeah, so you're talking about isolating the primary, so the cancer cells to use as instead of the cancer cell. We have thought about that. As you know, it's a bit hard to establish this in vitro, the primary cells. And within the timeline of the project, we felt that for now, we decided to test this initially with the GOT1 cells. But if we can manage to isolate these primary cells from patients, that would be an advantage, yes.
We're happy to send you protocols for the isolation.
Sorry?
We are happy to send you protocols for the isolation. It's not difficult.
That would be great, yes.
Okay,
Thank you.
Cool.
Thank you.
We'll invite Dr. Nakakura up next. Dr. Nakakura is from the University of California, San Francisco, and will be discussing their project on studying the microbiome of small intestinal neuroendocrine patients.
Thank you very much. I just want to clarify the work that I'm presenting today is from Dr. Netta Mackinnon, who's a postdoctoral fellow in Matthew Myerson's laboratory. She unfortunately could not be here today, so I'm presenting her talk. For the last several years, I've been very fortunate to collaborate with the teams of Matthew Myerson at the Broad Institute and Dr. Chrissy Thurwell now at Bristol Medical Center, trying to find out the causes of small intestine neuroendocrine tumors. And our approach has been to use high throughput sequence strategies to find the genomic as well as environmental causes of small intestinal neuroendocrine tumors. So this is the disease we're talking about. The prototypical small intestinal neuroendocrine tumor is a tumor of the ileum. In many patients, in some cases more than 50%, there's more than one primary tumor in the intestine. So we call them multifocal. They're usually located in the terminal ileum almost exclusively. And as we just heard from the last talk, Many patients develop lymph node metastasis, and around the lymph node metastasis, you get this very dramatic mesenteric fibrosis forming a mesenteric mass. The mesenteric mass causes kinking and buckling of the intestine. People are always amazed that these patients are always usually operated electively by me, and this is what they look like when we go in there. So this idea that they're asymptomatic is totally not true when you actually look at them. The bowel is usually ischemic partly. It's definitely contorted, and there's always a very high degree of partial small bowel obstruction. So the mesenteric mass is a big problem. Our sample cohort to address, to find the causes of this disease was to leverage our access to these multifocal ileum-NET patients. The initial cohort were 14 patients that had multifocal ileum neuroendocrine tumors. Many of them had associated lymph node and liver metastasis, as well as normal ileum and blood. Regarding the number of multifocal tumors, I think it ranged from 2 tumors in a patient up to 18 primary tumors in a patient. Giving background on what's known so far as the genomics of ileum neuroendocrine tumors, compared to other cancers, they have a very low somatic mutation rate. When you look at somatic alterations, the most common somatic alterations is chromosome 18 loss of heterozygosity, which is found in about 60% of patients. Years ago, I think at the NETRF, Matthew Myerson's group, as well as a group from Mayo Clinic, presented data that the only recurrent somatic mutation was found in CDKN1B, which was found in about 8 to 10% of patients. Other than that, no other recurrent somatic mutations have been identified. In the figure on the right is a study of over 2,500 solid metastases using whole genome sequencing, and you can see that neuroendocrine tumors had the lowest rate of candidate driver mutations out of all these cancers studied. So far, our collaboration has published 2 high sequencing papers that have, in summary, shown that these multifocal primary tumors are genetically unique. That is, they are not clonal. When you look at, on the left panel, patterns of chromosome 18 LOH, within a single patient, you can have different patterns. So you're losing different parts of chromosome 18 and from different alleles. So it's very interesting that these tumors are very different within the same patient. On the right, there's data to suggest that each tumor, each metastasis that you have has a potential to rise from a different primary tumor. That is, you don't know where that metastasis is coming from. It's not all just from the dominant or large tumor. It can come from different primary tumors. So it highlights the need that you need to find all these primary tumors and remove them because they each has a potential to metastasize. Because we didn't find any obvious recurrent somatic mutations other than the CDKN1P that was identified previously, and because the ileum is the main site of these tumors to arise, we hypothesized that maybe the tumor microenvironment might play a role in ileum-NET pathogenesis. In this study, we turned our lip to the microbiome. And there's a lot of evidence in other cancers that the microbiome may play a key role in carcinogenesis. We're all familiar with Helicobacter pylori in gastric cancer, hepatitis B and C, and hepatocellular carcinoma, human papillomavirus and cervical cancer, and Merkel cell carcinoma, which is a neuroendocrine tumor of the skin. There's integration of the Merkel cell polyomavirus. So there's a lot of precedents for the microbiome being linked to carcinogenesis. And given the fact that the GI tract is obviously very abundant in the microbiome and has a very diverse supply, it's an interesting target to evaluate. The technique we used was this computational tool called PathSeq, which was developed by the Matthew Myerson Laboratory. Basically, it's using high throughput sequencing data to identify microbes in human tissues. So let me just start with the general overview of how it works. You have your primary sequencing reads that you start with on the left. And then there's a filtering phase which we remove the low quality and repetitive sequences. And then any sequence that maps to human sequences is subtracted out. So that's a subtraction phase. And what you're left is with presumably non-human sequencing reads. And then those are then aligned to known microbial sequences. We studied over 144 samples. I shouldn't say we, Matthew Myerson's group studied 144 samples from 13 multifocal ileal patients. We also had 10 unifocal ileal neuroendocrine patients. And you can see the list of the samples there. Netta and Anders were two postdoctoral fellows in Matthew's lab that supervised Tong Gao, who was a summer student who did this work. So when we look at the major groups of bacterial microbial reads that were found in this tumor set, you can see that the bacterial reads were probably the most microbial reads that we identified in these patients. We found increased bacterial reads in the normal ileum, the primary tumors, as well as the metastases compared to the normal blood. Also of note, all that at a lower level, more fungal reads, viral reads, and eukaryotic reads were found in this patient cohort. The eukaryotic reads are single-cell organisms that are protozoa or archaea. One thing that they wanted me to point out is that data analysis of this set is ongoing. One of the key issues is eliminating contaminants. You would expect no bacterial reads to be found in whole blood, but as you can see here, well, you can't see here, but they did detect several bacteria in the normal blood in patients. So this needs to be addressed. These are the most prevalent phyla of the bacteria that were identified in this cohort. All of these phyla are bacteria that are found in the normal human gut. And as mentioned before, the top four phyla, you can see that in most of the patient's sample whole blood, these bacteria were also identified. So again, highlighting the need to what we call decontaminate or filter out potential contaminant bacterial reads. Now, this problem of decontamination of this kind of big data is a challenge in the field, and people are working on it and how to do it. But the challenge arises when you have a very low microbial biomass that you're starting with. You imagine like a human tumor isn't expected to have high amounts of bacterial reads in it. But this simulation shows that as you decrease the biomass of your microbiome, the problem of potential contaminant DNA increases, which is shown in the gray. So how you are able to filter out or decontaminate the data is a key thing. And at a very simple level, you can imagine whatever's in the blood you may want to try to subtract out, but there's statistical and methodological ways that this is going to be addressed. But once these things are addressed, all these questions that are on the right can be looked at. Do we find that there's any bacteria or other microbes that are enriched in the tumors compared to the normal blood and normal tissues? Is there any difference in the multifocal cohort versus the unifocal cohort? And do we see these also in metastasis? There's precedence in, like, for example, colon cancer with the fusobacterium being present at a much higher level in the colorectal cancer primary tumors and metastasis compared to normal tissues. So this is one potential hypothesis that maybe something like this is going on in ileal nets. Okay, to summarize that, at least so far, several microbial reads have been detected in our ileal net patient cohort. Most of these sequences come from bacterial, and these bacteria that we've identified were the ones that you typically find in the human gut. Importantly, we need to decontaminate the data, remove potential contaminants so we can see what is really potentially enriched in the tumor tissues compared to normal blood. And Just want to acknowledge the work was done in Matthew Meyerson's group by Netta and colleagues. Also want to acknowledge the ongoing collaboration with Chrissy Thirlwell's group at Bristol Medical School, as well as all the folks at UCSF. Thank you very much.
Usually the ileum actually doesn't have any bacteria, so it's considered to be a sterile organ. That doesn't mean there are any, but certainly a very small number. So when we talk about the human gut microbiome, we talk about colon, large intestine. How do you reconcile that finding? So you're referring to the human gut microbiome data.
I would disagree. I wouldn't say the ileum's sterile. I mean, there's a lot of bacteria in the ileum. I mean, it's not a magical decontamination process from going to the ileum. The ileum's only about this far from the colon, right? And the ileocecal valve's not watertight, so I would say it's not sterile. But one thing to keep in mind that they can detect bacterial reads even in blood, and some are not necessarily. Not all of them are contaminants. So this just shows you the very sensitive nature of this sequencing technology. They're looking for sequencing readings of very small quantities of DNA. And this is why there's a paper that someone recently published about this decontamination issue, is that people have tried to look at the microbiome based on published databases like in the Cancer Genome Atlas Project. And they were able to identify, again, contaminants in the blood that were bacteria that were in the blood that proved out to be contaminants. And they tracked it down to it tended to be site dependent. So certain sites had introduced contamination either from the sample preparation, the reagents. There's even talk about how what kit you use to purify the DNA can introduce microbial contaminants. So this is a very sensitive technology that can detect very, very small quantities of microbes.
Great talk, congrats. Just like curiosity, do you have data regarding the use of antibiotics or probiotics in the days or weeks before the surgery?
Yeah, I mean, routinely for most of these cases, at least at our institution, we'll give oral antibiotic and laxative before the surgery, but also just before the incision, we'll give intravenous ceftriaxone and metronidazole.
Yeah, and do you think that might skew the results in any way?
Yeah, I mean, it may. What's interesting, well, I don't think it's skew if it's a virus, right? If it's an integrated virus, would it skew it if it's if you look at the fusobacterium data in colorectal cancer? They find such high elevated reads in the cancer samples themselves, in the cytosol of the tumors. So that I don't see how that's going to affect that necessarily, but I mean, it may.
I just have one quick question. Hi, Dave. So I'm one of the Surgeon Fellows from MD Anderson. I saw that you also analyzed the metastatic disease for the same microbial reads. And it looked like there was a lot of bacterial signal in there. Are these mostly liver mets?
A combination of liver and lymph node metastases.
Gotcha. And is your thought that the bacteria sort of travel with the metastasis from the primary site, or is it sort of colonization of the secondary?
Yeah, looking at the data that links fusobacterium to colorectal cancer, they find that the bacteria is actually in the cytosol of the tumor cells. So it's actually in the primary and also in the metastasis in the cytosol of the Mets. So it's not like bacteria is just sitting on the tumor and going with it. It's something more complex going on. And there's a whole, as you saw in that review, there's a whole body of literature of people evaluating how the bacteria interacts with different hallmarks of cancer, from uncontrolled growth signals to invasion, things like that.
Yeah.
So there's a lot of potential mechanisms.
It'd be really interesting to see if the paired sort of primary versus met within each patient, how much concordance there is in terms of the bacterial phyla that are present. But thank you so much.
Yeah, no, those are good questions. Yeah.
Hey, Eric, Matt did this analysis.
So Chris, maybe we can save that for the panel discussion. We should. No. If we could please. You don't have to answer that question. No, Chris, we should have the conversation, but let's allow the next speaker to have their turn.
And if our first speakers want to come in.
And start occupying the days to our left. We'll continue the topic of the microbiome with the last speaker, Marian Mulders, talking about mid-gut neuroendocrine tumors and gut microbiome.
Yeah, hopefully it works. Yeah, there we go. I'm happy to see that I'm not the only one doing microbiome research in neuroendocrine tumors. I'm Marijn Mulders. I'm a PhD student from the Erasmus Medical Center in the Netherlands, which you see here. And I'm happy to share you the details about my research regarding the role of the gut microbiome in mid-gut neuroendocrine tumors. As of recently, we're able to identify which microbes inhabit the human gut with advanced DNA sequencing techniques. These microbes have been shown as relevant modulators of tumor behavior across a wide variety of types of cancer, including gastrointestinal cancer. And more microbes and microbial metabolites have been shown to be novel treatment targets. And in addition, the enterochromaffin cells of the gut can be stimulated by microbial metabolites, as shown here in this graphical abstract of a paper published in Cells. So because mid-gut neuroendocrine tumors derive from the enterochromaffin cells, the neuroendocrine cells of the gut, we chose to study the gut microbiome in mid-gut neuroendocrine tumors. More than 90% of the total body serotonin is synthesized by the enterochromaffin cells of the gut. And serotonin is the main mediator of the carcinoid syndrome, the most prevalent hormonal syndrome in neuroendocrine tumor patients, prevalent in about 20% of all patients. These patients have symptoms of diarrhea, vasomotor flushes, which you see here, bronchospasms, and fibrotic complications. And due to the median survival of patients with mid-gut neuroendocrine tumors of about 5 to 10 years, these patients have these symptoms for prolonged periods of time, severely impairing their quality of life. And they have limited treatment options, especially when they're refractory to somatostatin analogs. So improved understanding of the carcinoid syndrome is urgently needed. So our aims are therefore to map the gut microbiome of mid-gut neuroendocrine tumor patients and to compare microbial composition of patients and controls, and patients with and patients without carcinoid syndrome. We did this by fecal sample collection and collected relevant metadata from questionnaires and from electronic health records. And we did 16 srRNA sequencing and whole metagenome sequencing. And we performed 4 types of analysis on this data. First is alpha diversity, where you simply count the bacteria that are present in each sample. Second is beta diversity, where you compare microbial composition between your 2 groups. And third is differential abundance analysis, where you identify several microbial species that are more or less abundant between your groups. And the fourth is a machine learning technique we use to create a microbial signature that's able to discriminate between 2 groups. So we did this on 87 mid-gut neuroendocrine tumor patients. We excluded patients with appendix neuroendocrine tumor origin, and we excluded patients that used antibiotics within 3 months of sample date. And 53 of these patients had the carcinoid syndrome, 34 did not have the carcinoid syndrome, and we included 95 controls. And these were people that lived in the same household as the patient. And in a subset of this cohort, we did whole metagenome sequencing. These were 30 patients with carcinoid syndrome, 30 without carcinoid syndrome, and 20 controls. So first, when comparing the counts of bacteria, we see a clear difference between our 2 groups. We see that the patients, which each dot represents a patient on the right side, have a lower bacterial count compared to controls. And this difference was statistically significant. There are also other measures of alpha diversity, where you also take the spread, the evenness into account of these bacteria in the sample. And again, we saw similar results. We also corrected for relevant confounders with a linear regression model, including certain types of medication and previous GI surgery, And again, we saw a statistically significant difference. Then we looked at beta diversity. And this is a multidimensional analysis here captured in a two-dimensional plot. And we see that the purple dots, which are the patients, cluster around a different point than the orange dots, which are the controls. And again, we see a statistically significant difference, and we corrected for the same covariates as in the alpha diversity analysis. And then we identified several differentially abundant species. We did this for a 16S rRNA analysis, and we identified 31 differentially abundant species, which overlapped in 4 different statistical methods. And here on the right side are the results of the metagenomic differential abundance analysis, which we did with 2 different statistical methods. And we see that the dots are the bacteria which were identified. And if the dot is on the right side of this volcano plot, it's enriched in patients. And if it's on the left side of this volcano plot, it's enriched in controls. And I would like to highlight 2 bacterial species of this analysis. The first is the Veillonella atypica, and this bacteria has also been associated to pancreatic cancer. And another species of this genus, the Veillonella parvula, has been shown to drive oncogenesis in lung cancer models. The second is the Faecalibacterium species. The most known is the Faecalibacterium prausnitzii, which is a butyrate-producing bacteria. And sodium butyrate has shown potent anti-cancer activity as a histone deacetylase inhibitor. And ingestion of sodium butyrate can induce apoptosis in colorectal cancer models and inhibit tumor growth in lung cancer models. And then we did a 10 times 10-fold cross-validation with the random forest machine learning technique, and the results of which are shown here in this heat map. And you see that the signature discriminates between patients and controls with high discriminative performance, with an area under the curve of 0.86. The top 6 bacteria on the right side are the ones that are enriched in samples of patients, and the bottom 11 are the ones that are enriched in the samples of controls. And as shown by these results and results of other studies, many disease states seem to have their own specific microbial signature, and identification of such a signature could lead to early disease detection and perhaps more chance of curative treatment. But global multicenter collaboration is needed to validate these findings and to implement them into patient care, and to make sure that there's no geographical bacteria confounding these results. And then we looked at patients with and without carcinoid syndrome, and to our surprise, we did not see any difference in all of the analysis. So alpha diversity, beta diversity did not show significant results. We did not find any differentially abundant bacteria, and the area under the curve of our microbial signature was 0.5. However, we were able to discriminate between patients with carcinoid syndrome and without carcinoid syndrome based on the bacterial pathways that were active. And this pathway had an area under the curve of 0.8, this signature, and this could constitute a novel biomarker for the carcinoid syndrome. And then we looked at which bacterial pathways were more or less abundant in the patients versus controls analysis, and we saw again several differentially abundant pathways, and several of which have been linked to drive cancer in other cancers. So this is an interesting way of looking at this, but these findings and the findings of the differentially abundant bacterial species are purely associative, so they need to be validated with in vitro and in vivo work. And then we did several subgroup analysis. First, we looked at if there was an effect of tumor multifocality, and we did not see differences in microbial composition of patients with multifocal tumors and without multifocal tumors. We also did not see differences in patients with grade 2 tumors versus grade 1 tumors. And then we questioned whether somatostatin analogs could have confounded our results. But when we compared patients that did not use somatostatin analogs to controls, we saw similar statistically significant differences. So to conclude, mid-gut neuroendocrine tumor patients have an altered gut microbiome. It was less rich and less diverse than controls. They have a different microbial composition. Several differentially abundant species and pathways were identified, and we created a discriminatory microbial signature. And this difference in microbial composition seems to be independent of the presence of the carcinoid syndrome. But it looks like these findings suggest a role of the gut microbiome in neuroendocrine tumor development. Several potentially protective and oncogenic mechanisms have been identified and can be used for follow-up studies and screening patients for a microbial signature can be the future of early disease detection, but I think that global multi-center collaboration is needed to implement these into patient care. So with that, I would like to thank everyone that has worked on this project, and also NETRF for making this project possible and allowing me to present my work. And with that, I would like to thank you for your attention, and I'm happy to take any questions. Yes, so it's in some patients they have received somatostatin analogs and some didn't, and then we corrected for that in the subgroup analysis. But also some patients received previous the gastrointestinal surgery and some didn't, and we corrected for in a linear regression model. So it's mixed, yeah. Yeah, we did that with the somatostatin analogs, like patients that did not use somatostatin analogs versus controls. But
thank you. That's really, really interesting work. We've seen in many other cancers that the microbiome has a really significant impact in response to different therapies, for example, immunotherapy and others. And I just wondered what plans you might have to look at that in our various NET populations on treatment.
Thank you. I think it would be really interesting to study the microbiome before and after start of somatostatin analogs and see what the changes are, and like maybe then correlate that to which patients do respond, which patients do not respond. So before and after starting treatment would be interesting also for example, PRRT or something like that. Yeah.
And do you have plans to do that already?
Yeah, we have plans to do that,
yeah.
But yeah, I'm happy to, if anyone's doing microbiome research, I'm happy to collaborate.
Brilliant, thank you.
Thanks. I just want to drill on this one point for a moment, though. So for the carcinoid syndrome comparisons, were the patients clinically under control with their syndrome, such as by somatostatin analogs, when you evaluated for a difference in the microbiome, or were they prior to the establishment of clinical control?
So the majority of patients were under control with their symptoms. And we also looked at the subgroup analysis where patients had so we looked at patients that had a 5-hydroxyindolic acetic acid level below 50 or above 50 at the time of sample date. And there we tried to make the comparison that patients who have really high 5-HIAA have a different microbiome than patients that have lower. And there we saw some differences. So perhaps the patients being under control confounded our carcinoid syndrome analysis a little bit.
Yeah, it's really nice work. It might be worth digging a little bit more. Yeah.
So maybe you can join the other speakers, and we can open it up for a general discussion of the whole session. Chris, would you like to? You had a question that we didn't have time to ask earlier. Others?
I have a quick question. This is another microbiome question, but this is very basic. This is just a chicken and the egg question. Does anybody have a sense as to whether the microbiome is altered as a consequence of having the neuroendocrine tumor, or whether it is a driver and it's actually facilitating the development of cancer?
With our research, we can say whether it's the chicken or the egg, but I think it's an important first step to take, and then you take that to in vitro and in vivo studies to study that relationship more closely. You saw it with the Fusobacterium nucleatum. They first identified differences, and then they took that further to like identifying a bacteria that inhibits reaction to chemotherapy.
And that's the critical question in this field: is it just an association, or is it causal? Right. I mean, you have a disease, the bacteria changes in the microbiome, right? Or is it the reverse? Right. You have to do those things. Yeah. But I'm curious, since the world's experts are here, what do people think is causing ill nets? I'm curious from taking a poll. Is it a virus that we haven't found? Like Merkel cell, like HPV.
This is great. I love it when the speaker is asking the questions in the audience. This is wonderful.
Got to take advantage of this. Got to take advantage of this. I can never get these people together again on a Zoom.
It's lost of the printing of IGF. It's present in 55%. Then there's another gene that's present in
The Wnt9.
You go to a microphone so we can all hear. You could question.
The question from Eric was about why these multifocal.
I mean, loss of imprinting of IGF-2 is found in 55% of patients, so I figure that's one of the triggers. And the other 45% or so seem to have a Wnt9b problem. So I think that's probably the first step. And then there's a lot of MDM2, which we're not quite sure why. So, and that drives down to P53, and that's why we can see P53 and IGF-2 driving siNETs in mice. So I think those are probably the first two things.
Hi, I'm wondering if I know you're. This is the small bowel neuroendocrine tumor panel, but have you compared like the microbiome signature from, for example, PNET versus SPNET, or is that in the pipeline soon?
Yeah, we are planning to do that, but I think it's also important in microbiome research to get like some PNET samples from this side, some PNET samples from that side, and then some lung net samples again from similar sites, and then compare those.
Yeah, and I just kind of touch it. Maybe it will help us answer the chicken and the egg question that Chrissy mentioned. So, for example, if there is a differential signature, then we can kind of like associate it. Either maybe is it like a specific hormone or neurotransmitter that is secreted by this specific type of neuroendocrine tumors.
Just a thought. We've got a list of bacteria that are at the top end of the chart for neuroendocrine ileal cancer. Might one of those bacteria be causing, or a group of them together, causing the cancer, and causing in addition, causing the carcinoid syndrome and the diarrhea and everything else that goes with it? I have no idea whether that's an avenue to explore or not, but when we have something that's visible, that's different, my initial instinct is to let's go look and see if that's where the smoking gun is, because it certainly smells like cordite to me, but that doesn't mean that it's cordite.
Yeah, I think that's a thing to look at. That's what our research question was, and why we performed the study, to see if there are special bacterial species that are causing neuroendocrine tumor cell growth or carcinoid syndrome. So yeah, things like the ones that we identified may be valid targets for follow-up stuff.
But I think the symptoms from carcinoid syndrome, like the flushing, diarrhea, are thought to be related to serotonin and other mediators that are released by the tumors once you get liver metastasis, distinct from bacteria causing diarrhea, for example. Yeah.
Eric, I wanted to ask you this question. So you, at the beginning of your talk, you mentioned that a lot of these patients with the small bowel disease have a lot of structural problems by the time you take it out, as far as they've had adhesions or whatever it looked like. Are there instances where this gets diagnosed really early by a colonoscopy or something, that before you have any structural problems in the bowel? And because it seems like that might be good to start testing early on. I worry, you worry that you're going to end up encouraging a lot of bacterial overgrowth and microbiome changes
if you wait too late. So I just wanted to see what your thinking on that was.
For sure, if you get it picked up early on colonoscopy, there's the handful of patients that get screening colonoscopy found a terminal ileal neuroendocrine tumor. And you're right, the mesentery is completely normal feeling. There's no lymph node metastases, no mesenteric mass. You're saying a study though, microbiome of those folks, yeah. I think it's hard. Yeah, I guess you'd pick those up. It's just there's not that many patients. I mean, they come along, but
right. But do they do surveillance screening by colonoscopy at all, or you just bring them to surgery if you see that? Because
yeah, we just take them to surgery. Yeah.
You're a surgeon. Just take them to surgery. Right?
That's it. Easy question.
Hi, I'm Hyan Lee. I'm from Fox Chase Cancer Center. This is also question or opinion. I'm actually wondering what kind of reference genome do you use to screen out human artifacts before you actually pursue microbiome or metagenomics or something?
The question is, how do you screen out human artifacts?
Yeah, screen out human. You said subtract the human genome.
Yeah, so the path-seq that Matthew Myerson's group described, and again, this is like me reading all these papers on the airplane over here. So I can tell you with that extensive knowledge. But the path-seq computational tool was developed by Matthew Myerson, I think, about 10 years ago, where you take the high throughput sequencing data, get all your reads. I told you filter out the low quality data, the repetitive sequences, the duplicate sequences, and then you see what sequences align to known human sequences. And they use several, like 4 or 5 databases, and then all those are taken out. I mean, you just take them out, and then supposedly you're left with the non-human reads. And then they take those non-human reads and then blast them or align them to known microbial reads. But you can see that if you have like a very small virus or something that's hiding in a hard to sequence area, and it's out of all that data, how it can be very hard to find. And that's,
Yeah. So basically that database, I think that's built up based on some reference genome, like the human reference of the HG 38, or the genome that lately built last year. Yeah, that's what I'm going to say. So basically it's not really updated so far. And even TCGA data is reference genome was HG 38 is used, which is assembled like 2004. And last year we have a new reference genome that we feel all repeat. So in fact, the previous genome has only 92% of assembled, and the last 8% of human reference genome is assembled the last year, which is almost repeat. So basically, if you want to remove human artifacts, the database should be also updated based on the latest human reference genome.
I can ask Netta and Matthew that to answer.
That's quite really important because the repeats, half of the repeats that newly assembled came from Y chromosome. So that makes actually a big difference between this. Your samples are processed by the male technician versus the female technician. Because sometimes it's human artifact, the skin dust can go into your samples, and then you think you removed all human artifacts from your samples or sequencing data, but you actually removed only 92% of human genome, and 8% are left there if you don't use the latest reference gene. So yeah, that's my point. So always, this is something that I reviewed the paper. People still use very old reference genome, sometimes HG19, which I think we should remove that one. But anyways, yes, it's really important for all of the research to use the latest updated human reference genome, and especially metagenomics. It's really, really, really important. Yeah, that's my.
Thank you. Maybe as long as we are on the microbiome topic, I'm struggling in thinking about this conceptually. So I can understand that a viral or some other pathogen genome might be integrated in the human chromosomes and would be detected by this method. But Eric, you referred to bacteria that are carried in the cytosol. And so operating on that presumption, Is there any simple back of the envelope calculation as to how many reads one might expect? You might say, okay, if there are 100 bacteria in a cell or 1,000 bacteria in relation to the 3 billion base pairs in the haploid human genome, what you might expect is the numbers seem awfully small. And picking up on the last comment, I just wonder if maybe the technician didn't wash their hands before they prepared the DNA. Then you would see it in the blood and the met as much as you would see it in the primary.
Yeah, so interestingly, surprisingly, like I mentioned, some of the contamination in prior studies have been linked to the DNA purification kits themselves. Like they find the bacteria in there, in the kit, used to isolate the DNA. So, I mean, it's not like people are being really sloppy. It's just a very sensitive technique. It's very sensitive. I had a question about the mesenteric mass work. Very nice work. As a surgeon, obviously, we have to take these things out. I was curious your thoughts of. I understand you're doing the cold culture experiments to see why they grow so big. Is your thought that that's why they're so big in the mesenteries? They have this unique environment that's promoting the growth. So the primary tumors and the liver mets aren't that big, but the mesenteric mass is big, and it's mainly fibrotic. Is that the thought?
Yeah, so the thought behind this is that the interaction between the fibroblast and the cancer cells is what's promoting both the fibrotic reaction and also the growth in the cancer cells.
Are those calves or fibroblasts not in the normal ileum and liver, and that's why the mesenteric mass is disproportionately big? Or what's so special about the mesentery?
So that is what we're trying to investigate. That's why we're also collecting from the normal tissue, the normal mesentery and the primary tumor, in order also to compare how these fibroblasts react and how they interact with the cancer cells. And that's what we want to model and study. Yeah.
And once you figure this out, what was the ultimate goal? Is to figure some way to prevent it or to reverse it, or?
Yeah. So the goal is to try and identify something that is targetable. Now we can use as a therapy. Of course, that we have to consider that a lot of these patients already have a significant mesenteric mass when they're diagnosed as well. But we're also trying to identify, as I said, biomarkers for detecting MF. And we have already done some studies on patient bloods that are published to identify mesenteric fibrosis also at initial stages of the disease. So if we can do that and combine that with the treatment, that might be beneficial for the patients.
And I guess it's often hard to tell where the fibrosis begins and the lymph node metastasis ends, or you could have like mainly lymph node metastasis, or is it mainly fibrosis? Because sometimes we send off a thing that's this big and they tell us there's 2 lymph nodes in there and it's all fibrosis, or they may say there's 10 lymph nodes in there and a little fibrosis. I'm just curious how you process it. Are you analyzing a lymph node met, for example, in one sample, and then in another one it's just purely fibrosis, and how do you account for that?
So these tissues are usually processed by our pathologist that is specialized in SINETs. So what we usually do for the biobanking, so the tissue that we analyze fresh frozen, and also when we isolate the fibroblasts, is taking the tumor, so it would be the lymph node and a bit of the surrounding fibrotic tissue as well. So we have a representation of both of these compartments. If you have fibroblasts also on the surrounding stroma compartments, they will also have an effect on the adjacent tumor cells.
I'm going to take the privilege of the last question here. I was wondering, when you're looking at the pathway for fibrosis in SINET, do you know if the pathway or the biomarkers that you're finding are similar to other fibrotic processes, particularly in DSFP, different sarcomas or idiopathic pulmonary fibrosis? Are there similarities, or is there something unique about this?
I would say that there are always kind of common genes in fibrosis that have been described in fibrosis from many different diseases that pop up, but we're trying also to focus on more specific and targeted genes that might be driving this specifically in mesenteric fibrosis. But yes, usually, as I said, for example, those 9 genes that we identified were already described in literature and not in SINET specifically, but in other conditions, like liver, for example. So yeah, some of them will be already described in other diseases as well as common pathways.
All right, I want to thank everybody for attending the session and all the excellent questions and discussion.